Very good morning. So, shall we start human physiology? And when I discuss these topics, mostly I think 90% of the points you can recollect. When I'm explaining automatically, plus you need to check yourself whether you are able to recollect all those points when we are discussing that, right? So it's a check, and again after this class, tomorrow or day after, when you are preparing, focus mainly on those points and try to complete each chapter within a short time. You can check the time, how much time we are taking to discuss. Okay, you also try to complete the same half an hour, one chapter, or based on the chapter. Like circulation, it may take more time, or respiration less time, it depends. So whatever it is, you also try to complete within a short time. That's the purpose of this class, how to focus and which points on we have to focus. Now coming to this human physiology, first let us start with breathing. Breathing, what is the main purpose of breathing, or what is the use of respiration? First of all, every living organism, so coming to this, every cell needs energy to perform its metabolic activities, and energy is released by oxidation of nutrients, that is glucose, amino acids, etc. And this oxidation needs continuous supply of oxygen, and when oxygen oxidizes the food, form carbon dioxide and water. Carbon dioxide is harmful when it accumulates inside our body. That means there is a need to eliminate carbon dioxide. So need to take oxygen and send out the unwanted or harmful carbon dioxide. That is the main purpose of breathing, taking in air and giving out. Okay. And in the introduction paragraph, we talk about various structures. The type of respiratory organs which help in this breathing depend on habitat and level of organization. Habitat and level of organization. First time, lower organisms, sponges, cnidarians, ctenobots, platyhelminthes, low level of organization, no special structures needed. It occurs by diffusion through body surface. And annelids are having moist cuticle for exchange of gases, and then arthropods, we see wide variety of structures because they are found in all habitats. Book gills, book lungs, gills. Remember all those structures. Okay. And aquatic arthropods and molluscans also have gills. Terrestrial molluscans are having vascular bags called tracheal system, is characteristic of insects. Mollusca also mostly aquatic and gills. Echinodermata, water vascular system helps in that process. Hemichordata, gills. Hemichordata, pharyngeal gills. And among chordates and vertebrates, lower organisms up to fishes, gill respiration. And amphibians onwards, lung respiration. Amphibians also use skin for respiration. Okay, that's about it. And coming to human respiration, it starts with, as our lungs are arranged in the thoracic cavity, we need a passage or pathway to carry the atmospheric air to the lungs. It starts with the pair of external nostrils. Common mistake you are making. After external nostril, is it nasal chamber or nasal passage? Through nasal passage, it opens into nasal chamber. And inside these nasal chambers, twisted bony plates are present called concave or turbinals. Three each side, superior, middle and inferior. And then these nasal chambers open through internal nostrils into pharynx. Pharynx, a part of this is common for both food and air. Food and air. Then pharynx, through an opening called glottis. Opening is glottis, leads into larynx, and as pharynx is common for both food and air, there must be separation. Entry of food or water at the time of swallowing is prevented by means of epiglottis. Nature. It is elastic cartilage, made up of elastic cartilage. It prevents the entry of contents into the lung. Larynx is commonly called as voice box, made up of cartilages. Total number of cartilages associated 9. and inside the larynx a pair of elastic cords are present. They are vocal cords. Vibration of vocal cords only produce the sound, and this sound produced is different in females. High pitch voice, males low pitch. Low pitch voice is due to large and thick vocal cords which occur due to androgens. At the age of puberty, the change in the voice occurs due to androgens, making the vocal cord thicken and larger. So they vibrate less, causing low pitch voice. Okay. And in addition to that, larynx followed by trachea. Trachea is commonly called windpipe. What is the other pipe running parallel? Esophagus or food pipe. Esophagus or food pipe. And which one is ventral in position? Trachea. Esophagus is back side. Trachea is towards ventral side. Okay. And it runs through the neck, thorax, mid-thoracic region. It bifurcates into two primary bronchi. Each primary bronchus enters the lung of that side and divides. There is a lot of branching inside that. Primary divides to form secondary. Tertiary bronchi. Tertiary bronchi divide to form primary, secondary, tertiary, terminal bronchioles. Terminal bronchioles end with respiratory bronchioles and alveolar ducts, alveolar sac ending with alveoli. Actually, in NCERT, respiratory bronchioles, alveolar ducts are not mentioned. They simply use the word alveoli and their ducts. And this part constitutes exchange part. Actual exchange of gases take place at this site. Starting from external nostrils to terminal bronchioles is conducting part, and this helps to transport the air towards the alveolar region and humidifies the air, filters the foreign particles or dust particles, and also brings the temperature of air equal to body temperature. That is the function of this conducting zone and exchange zone, right? And this whole branching constitutes the lung. Lungs are the thoracic cavity and towards left side. Yeah, heart is tilted slightly towards left side. That's why left lung is having cardiac notch, a small depression due to the location of that. And lung is enveloped by double-layered pleura with the pleural fluid, and it reduces friction near the lung surface. You know about it. And outer pleural layer, it forms lining for thoracic, inner surface of the lung. And lungs are arranged inside the thoracic cavity in such a manner, it's airtight chamber in such a manner that any change in the thoracic volume directly reflects on lung volume. If thoracic volume increases, lung volume also increases. If it decreases, directly proportional. And thoracic cavity is bounded mid-ventrally by sternum, dorsally by, laterally by, and lower side by muscular partition diaphragm. Okay, so this is, and last point regarding this is this kind of arrangement of lungs in the airtight thoracic chamber, which directly, why is it needed? Why is it essential? Because we cannot directly alter the lung volume. There is no musculature inside the lung that we can control. But we need to make changes in the volume of the lung. That's why lungs are arranged in such a manner so that we can alter thoracic volume by means of muscles. Okay, which muscles? Diaphragm and intercostal muscles. So all this is about that. And here one more important point. Trachea, bronchi and initial bronchioles, they are supported by complete or incomplete? Incomplete. What type of cartilage? Hyaline cartilage. Hyaline, not elastic. Hyaline cartilage. They prevent the tubes from collapsing. They give support to the tubes. Okay. And then alveoli are lined by simple squamous epithelium. That's why it is very much suited for diffusion or exchange of gases. And coming to these steps, literally all these 5 steps are represented in that diagram. What is the first step? Breathing. Breathing is nothing but ventilation. which is nothing but movement of air in and out of the lungs. So in this diagram, you can see all these 5 steps. First one, movement of air, inward and outward, that is between atmospheric air and the lungs. What is this step? Breathing or pulmonary ventilation. That is the first step. Okay. And now there is exchange of gases between alveolar air and blood reaching the lung. What is this called? Exchange of gases occurring in lungs. Pulmonary exchange. And now it is followed by transport. Oxygen from lungs to tissues. Carbon dioxide from tissues to. This is the third phase called transport of gases. Fourth one, after reaching the tissues, again exchange of gases occur between blood and tissues. This exchange is called systemic exchange or external, internal, pulmonary systemic exchanges. And the last step is utilization of oxygen by the cells forming the carbon dioxide with the release of energy. This is called cellular respiration and occurs inside the cell, and that is discussed under separate chapter, respiration. So we need to discuss these 4. First one, breathing. Exchange of gases we discuss simultaneously, both exchanges and the transport of gases. Okay. So first one, breathing. Breathing or ventilation, it involves 2 steps. What are? Expiration. And this is facilitated by pressure gradients, because gases always move from higher to lower. So pressure gradient is needed for this process to occur, and these gradients are generated by special muscles, diaphragm muscles, also called phrenic muscles, and intercostal muscles, which extend in between the ribs. Okay. And always intrapulmonary pressure must be less for inspiration to occur than outside. Same manner, when intrapulmonary pressure exceeds than that of atmospheric, expiration always from high to low. And this, we cannot alter atmospheric pressure, we can alter only. So we can make this pressure less or more compared with that with the help of these muscles. And the process is initiated by first. What is the shape of this? Contracts and it becomes, you can say flattened or you can say pulled down, whatever it is, pulled down or flattened, which causes increase in the thoracic volume. Which direction? Anterope. Because 2 different axes, this causes change in longitudinal or anteroposterior axis. Volume increase or decrease? Increase. Same time, external intercostals also contract, due to which rib cage is raised up, pulled outward. You can say outwards, upwards, both are correct. Okay, like this. And this causes change in the volume dorsoventral axis. That means both the axes volume increased. Which volume? Thoracic volume. Thoracic volume change is reflected. Pulmonary. So pulmonary volume also increases, due to which intrapulmonary pressure decreases, becomes less than outside. So outside it rushes into the, this is inspiration. How do you describe this? Active or passive? Because it occurs by contractions of muscles. So it is active process. Done. And then after the exchange of gases, expiration occurs. Reverse, nothing special. These 2 sets of muscles which underwent contraction earlier, now they become with the relaxation. Diaphragm is again arched upwards or becomes dome shaped or regains its original. Same manner, rib cage also moves back to original. Check the diagrams when you are learning. Inspiration, expiration diagrams are there. Okay. And with this, thoracic volume decreases both the axes. With that, pulmonary volume decreases, intrapulmonary pressure increases, exceeds than that of outside, and air rushes out. This is expiration. Normal expiration is just caused by relaxation of muscles, so it can be described as passive process. Inspiration, active. Whether it is normal inspiration or forced inspiration, it is always active. Okay. Normal expiration is passive, but forced expiration occurs by contraction of internal intercostal or additional muscles. That's why you can describe that as active, unless they specify forcible, don't. Okay, depending on the statement. And what is the rate? 12 to 16 times. rate of respiration or breathing is 12 to 16 times, and we have the ability to alter this with the help of additional muscles. And what is the instrument to measure? Spirometer. And here we are going to discuss the volumes and capacities. Volume of air, I am asking questions. Volume of air remaining in the lungs even after forcible expiration. Residual? 1100 to 1200. Earlier book additional volume word is there. Now it is additional word is deleted. Volume of air that can be inhaled by forcible inspiration is called as IRV, inspiratory reserve volume. 2500 to same definition expiration ERV by forcible expiration ERV, that is 1000 to 1100. Volume of air moving in and out, the normal breathing is called, that is one breathing. Per 1 minute it would be 6, 12, because we breathe 12 to 16 times, multiplying that you get 6000 to 8000. And combining the volumes we get total volume of air a person can expire after normal inspiration. Easy. See, after normal inspiration, easy. TB plus? Yeah. Volume of total volume that a person can inspire after normal expiration. IC. TB plus? Maximum inhaled after forced expiration. Vital capacity. Other way you can say maximum exhale after forcible inspiration. This is called VC. TB plus IRV plus ERV. Total volume of air accommodated in the lungs at the end of forcible inspiration is called total lung capacity, TLC. It is, wait, VC plus RV or IC plus FRC or TB plus IRV plus ERV plus RV. Which volume cannot be measured by spirometer? Residual volume. Done with that? So that is about it. Next topic. Exchange of gases, we have to discuss 3 points. Because exchange of O2 and CO2, it is facilitated by, one is pressure or concentration gradient, second one solubility of the gases, third one thickness of the membrane. Okay, 3 points. First one, pressure or concentration gradient. If you consider the pressure or concentration gradients, what is partial pressure? Pressure exerted by individual gas. Total atmospheric pressure is called as partial pressure, and you know how to calculate PO2. What is the percentage of oxygen in the atmospheric air? Approximately 21%. Atmospheric pressure is 760 mm of Hg. So 21% of 760. You get that value. Okay. Remember the data? O2, PO2, PCO2 in atmospheric air. 159 and 0.3. Alveoli 104, 40. Oxygenated blood. And which vessels you see oxygenated blood? Main ones. Pulmonary vein, systemic arteries. Deoxygenated blood and tissues both have 40 and 45. That's the numerical data. And now look at this one. At the alveoli, PO2 is 104 and PCO2 is 40. What is the blood reaching the lungs? Deoxygenated. Deoxygenated blood values are 40 and 45. That means there is a gradient for oxygen from alveoli into blood. Carbon dioxide 45 and 40. That means gradient for carbon dioxide is from blood to. Okay, automatically exchange occurs. Due to this exchange, blood becomes. And on reaching the tissues, now this is systemic exchange. Here, 95 and 40. 40 and 45. So, gradient for oxygen is from blood into CO2 from. And blood becomes. And returned back to the lungs. So, if you consider both the steps put together, that is given in the book also, there is a concentration gradient for oxygen from alveoli into blood and then from blood into ultimately oxygen reaches the tissues. Same manner, gradient for carbon dioxide. CO2 is formed in the tissue, starts here. Gradient is from tissue to blood and from blood to, so ultimately CO2 is sent out from the alveoli. Is that clear? This is the pulmonary exchange and this is the systemic exchange. Both exchanges done. And here we discuss exchange with the help of pressure or concentration gradient. Second point is solubility. So if you consider solubility, carbon dioxide solubility is 20 to 25 times than the oxygen. That's why there is a statement. The amount of carbon dioxide that can diffuse across the diffusion membrane per unit pressure difference is much higher for carbon dioxide than relative to oxygen. Okay, so it can readily diffuse across that. Is that clear? So solubility is one factor. And the next factor, thickness of the membrane. And thickness of the membrane, how many layered? Three. Three layers. How many cellular layers? Remember that. And the three layers are alveolar epithelium, basement substance, and endothelium of blood capillaries. Actually, both epithelia, squamous epithelia only, and epithelia always rest on basement membrane. That means between the two cellular layers, basement substance of both the epithelia are present. Don't count separately. Basement substance together as one layer. So totally three layered thing. How many cellular layers? Two. Direction of carbon dioxide diffusion. You need to start from endothelium, basement substance, and if they ask oxygen diffusion from alveolar epithelium, remember that point? Yeah, that's all. Overall thickness of this membrane is less than. So that is about it. What is the next one? Transport of gases. If you consider transport of gases, first one, oxygen. Oxygen is transported by the blood in two forms. 97% transported. RBC bound to hemoglobin as oxyhemoglobin. Only 3% plasma. Because solubility of oxygen is less, only 3% is transported, dissolved condition. And regarding the transport of oxygen, this binding of oxygen to hemoglobin primarily related to partial pressure of oxygen at high pO2 readily combines with that to form oxyhemoglobin. Oxyhemoglobin. This is oxygenation, not oxidation. Okay. And is it reversible binding or irreversible binding? Not only this, oxygen, carbon dioxide transport, wherever, whatever reactions we discuss, all are reversible. Binding occurs at one place, dissociation occurs at the place. Either for oxygen or carbon dioxide. All are reversible reactions. And this binding depends upon PO2. At lungs where PO2 is high, readily binds to form oxyhemoglobin. On reaching the tissues where PO2 is less, reverse reaction occurs or dissociation of oxygen occurs from hemoglobin. Is that clear? And each hemoglobin is a tetramer with 2 alpha and 2 beta. Each globin chain has heme group with ferric or ferrous. Ferrous. It can bind with one oxygen, meaning one hemoglobin molecule can carry maximum 4 oxygen molecules. 75% saturated means 3. 50% 2. Maximal 4 can be plus 1 gram of hemoglobin can transport 1.34 ml of oxygen. 100 ml of blood contains approximately 15 grams of hemoglobin, which can carry approximately 20 ml or 19.4 ml of oxygen. Remember that data? And coming to the other points. Though PO2 is the primary factor which affects the binding, this binding is influenced by other factors. PCO2, temperature, H plus concentration. And you know, instead of H plus, they can use the word pH. And these two must be opposite. H plus less means H plus more means or acidity also can be used. More H plus means more acidity, low pH value. Okay, just relate if they use those words to frame the question. And now coming back, the effect of these factors on binding of oxygen to hemoglobin can be studied by this graph. It is called oxygen dissociation curve. This side we take partial pressure of oxygen, percentage saturation, and under normal condition when we plot the graph, what's the shape? Sigmoid, which indicates saturation increases very rapidly initially, slows down and becomes constant. After that, even the PO2 is increased, it cannot, because maximally saturated. Okay, and in oxygenated blend, what is the PO2? 95, 95, right? At that nearly 97% of hemoglobin is saturated. Deoxygenated blood, what is the value? 40. At 40 mm of Hg, percentage, last time they asked a question. 70, 70, 75. So 75% saturation. That means deoxygenated blood is still carrying 75% oxygen, which acts as a reservoir. That means if any tissue is having high metabolic activity, it can utilize that oxygen, extra oxygen. And this is a normal curve, that is not the normal condition. And the effect of other factors like temperature, H plus concentration, and PCO2, not only that, 2,3 BPG, all these can be studied using this graph. Whenever there is increase in these conditions or factors, curve shifts to right side. Shift of the curve towards right side indicates. I asked, what happened to percentage saturation? What happened to dissociation? What happened to amount of oxyhemoglobin? Because dissociation increases. Curve shifts to right side. And opposite set of condition, low temperature, low H plus and low PCO. The curve shifts to left side. Curve shifting to left side means more saturation. And what is P50? Definition. Partial pressure of oxygen at which 50% of hemoglobin is saturated is called as P50. And if you consider 50% saturation means here, right? 50% saturation. It occurs around 20, this is 30, so 26.7 something. Okay, that is about it. And next one. All the factors at the lungs favorable for formation of oxyhemoglobin, because PO2 is high, PCO2 temperature all are less. Reaching the tissues where PCO2 temperature H plus high, PO2 is less, all are favorable for dissociation of. Next one. 100 ml of oxygenated blood delivers. Always under normal physiological conditions. Suppose metabolic activity is high, more O2 is used up in the tissues, and in that case, more dissociation occurs. With fall in PO2, dissociation increases, or with raise in CO2 not. So definitely it is not always the same. Normal conditions 5 ml, but it can be increased or decreased depending on the condition. Then coming to transport of carbon dioxide, how many ways it is transported? 3 ways. How much is dissolved in plasma? 7%. It is more compared with oxygen because of greater solubility. 20 to 25% RBC in the form of carbamino hemoglobin. Carbon dioxide binds to amino group of the hemoglobin. Carboxyhemoglobin is formed by carbon monoxide, not carbon dioxide. Okay, and here 2 points are important. Where is carbon dioxide formed? Tissues. Now it is to be transferred from tissues to at the tissues where PO2 is less, PCO2 is high. Carbon dioxide readily binds to form carbaminohemoglobin. In that form, it reaches the lung, and at the lungs, where opposite PO2 is, PCO2 is, carbon dioxide dissociates from carbaminohemoglobin and released into alveoli to be sent out. Done. So they ask factors promoting dissociation of carbon dioxide or formation of carbaminohemoglobin. Okay. Then 70% is transported. Bicarbonates. Bicarbonate formation occurs inside the in the blood only. And look at this reaction. They asked question. Carbon dioxide combines with water to form carbonic acid, which dissociates immediately into bicarbonates and H+. That means formation of carbonic acid, dissociation of carbonic acid, both are catalyzed by carbonic acid. Not only forward reactions, reverse reactions also catalyzed by same enzyme. This enzyme is one of the fastest enzyme present in our RBC, abundant in RBC, negligible or small amount is present in blood. That means most of these reactions occur in the RBC. It doesn't mean absent in plasma. Okay, most occurs in RBC. And in the RBC, if you remember this chloride shift, the reactions occur, and with the reaction, what is formed? H plus and bicarbonate ions. Cell membrane is permeable to negatively charged but not to positive. So there is increase in the concentration of these ions because of the reaction. H plus cannot diffuse out. It is readily accepted by hemoglobin, forming reduced hemoglobin. Hemoglobin, by accepting H plus, acts as buffer, and it is rich in which amino acid? Histidine. Histamine is different. Inflammatory mediator, that is histamine. This is histidine, amino acid. And so H plus ions are taken care by hemoglobin. Bicarbonate ion concentration increases. So membrane is permeable. They start diffusing out into plasma. To maintain electrical neutrality between plasma and RBC, same number of chloride ions diffuse into the RBC. This is called chloride shift or Hamburger's phenomena. That means now bicarbonates are transported both inside RBC as well as plasma. In that form reaches the lungs. At the lungs, where PO2 is very high, affinity of hemoglobin to oxygen increases. So hemoglobin binds with that oxygen and releases H plus. H plus bicarbonate combine to form H2CO3, which dissociates into carbon dioxide, diffuses into alveoli to be sent out. And with bicarbonates getting converted back to carbon dioxide, bicarbonates which moved out earlier start moving in, sending chloride again. This is called reverse chloride shift. And here we talk about 100 ml of deoxygenated blood delivers to alveoli, not to tissues. To alveoli. Oxygen delivered to tissue. CO2 to be delivered to alveoli. And influence of carbon dioxide and H plus on binding of hemoglobin to oxygen is called Bohr effect. Reverse. Influence of oxygen binding of hemoglobin to carbon dioxide is called Haldane effect. Done? Next one. Next. Regulation. Regulation, 3 points are there. First one. We have the ability to alter the breathing rate. It is done by RRC, Respiratory Rhythm Center located in medulla. Always signals from that alter the breathing rate. And another center, pneumotaxis, located in form, that can moderate the function of RRC. When signals are given from pneumotaxic center to RRC, it causes decrease the duration of inspiration. When duration decreases, rate increases. Thus it can alter. And last part, chemical sensory structures or receptors. In the medulla near RRC, there is a chemosensitive area. Aortic arches and carotid arteries, they contain chemoreceptors. Chemoreceptors, chemosensitive area, both are sensitive to carbon dioxide and H plus ion. So whenever there is increase in these levels, immediately they detect and send the signal to RRC to make necessary adjustments in the breath. And then we have disorders. Asthma is even allergy reaction also. It is characterized by difficulty in breathing with typical wheezing sound, inflammation of bronchi and bronchioles. Emphysema common cause, cigarette smoke. Walls of alveoli damage. Respiratory surface area decreases. Number of alveoli decrease. Size of alveoli may increase. They become less elastic, more fibrous. In emphysema patient, which volume increases? RV increases. Others decrease, RV actually increases because lungs are not able to force out the air. And the next one. Occupational disorders due to continuous exposure to lot of dust generated in these quarries, mining and all those places. Our defense mechanism is not able to cope up, that is to filter out those dust particles, and gradually they start getting deposited inside the lung, leading to inflammation and fibrosis. Example, silicosis, asbestosis, siderosis, black lung disease, all those. Okay, next one. Body fluids. And actually introduction, what is the need for these substances? In our body, nutrients must be transported from intestine to all body parts, waste materials to excretory organs, gases from lungs to body parts. Transport is needed. And in lower organisms, sponges and cnidarians, what do? They use surrounding water to circulate their body, which helps in transport of substances. And first time special circulatory system appeared, Annelida. Most of the higher animals, they use blood and lymph fluids for circulation. First we go. Which one? Blood. Ask questions? Just randomly, because I think you remember all those points. Blood is also fluid connective tissue. Matrix is in fluid state, called as plasma. Percentage. Formed elements. Plasma has 90 to 92% proteins. And most abundant protein, albumin, helps in osmotic pressure. Globulins help in defense mechanism. Fibrinogen, prothrombin, clotting. Many clotting factors are there, but they are inactive. That's why normally blood does not clot. At the time of injury, when they get activated, clotting occurs. And then many minerals, calcium, magnesium, all those are, and all the nutrients, waste materials are also in transit. And now coming to the formed elements, in adults, hemopoiesis occurring. Bone marrow, the three types of formed elements, which is the most abundant? RBC. Next, platelet. Last, WBC. RBC are erythrocytes. Count 5 to 5.5 billion. Shape, biconcave, circular disc. Majority of the mammals enucleated. Advantage of biconcave shape, more surface area to volume rate, so more exchange of gases. Absence of nucleus and cell organelles help to accommodate more amount of hemoglobin, so they can transport more amount of oxygen. So that is about it. And lifespan of RBC, main function of RBC is transport of gases. And graveyard of RBC, after lifespan is completed, they get destroyed there. And next one, WBC, count 6,000 to 8,000. Short-lived cells, and these are the only nucleated cells in our blood, only nucleated cells, and they have the ability to come out of blood capillaries into surrounding tissue by a process called diaphoresis. And these are classified into two based on presence or absence of specific granules in the cytoplasm. And among granulocytes, least number, basophils. Nucleus is irregularly lobed, and they stain with basic dyes. They produce histamine, serotonin, etc. Play important role in. Eosinophils percentage 2 to 3%. And nucleus is bilobe. Very specific diagram clearly given in NCERT also. Bilobe, and they play important role in resisting allergy and infections, parasitic infections. And the granules stained with the acidic dyes. Eosinophils are eosinophils. So eosinophilia is increased count of eosinophils which occurs at the time of parasitic infections or allergy reactions. Neutrophils percentage 60 to 65%. Nucleus is multi-lobe, polymorphonuclear condition because it can be 2, 3, 4, 5 lobed condition. Granules stained with neutral dyes. These are phagocytic cells, act as microscopic policemen. And then among agranulocytes, large number lymphocytes, 20 to 25%. Two types. B lymphocytes are provide humoral immunity and cell mediated. So mainly concerned with immune reactions. Monocytes, 6 to 8%, highly phagocytic, largest WBC. Know that? Of all these, large sized ones are monocytes. Small sized ones are lymphocytes. Not number, size. I am talking about size. Okay. And the only cells which can again re-enter the blood circulation after diapedesis, lymphocytes only. Okay. And phagocytic agranulocytes, phagocytic granulocytes, platelets count 1.5 to 3.5 lakhs. Lifespan short, 5 to 9 days like that. And they produce many clotting factors, play important role. Fall in the count of this can lead to clotting disorders. They are formed from megakaryocytes of bone marrow by fragmentation. Next one. Blood. Blood groups. Blood groups are based on presence or absence of two antigens. Antigen is any substance which can induce immune reaction, and it can be protein, sugar, any substance. A and B antigens are sugars. Both are sugar molecules, and these are present on surface of RBC. Correspondingly, plasma contains antibodies. Two types of antigens, two types of antibodies. Anti-A, anti-B. Blood group is based on type of antigen, not antibody. Type of antigen. So those with A antigen are called A group. They have anti-B. They cannot have anti-A because A is self-antigen. And B antigen, anti-A. You know that. AB group cannot have antibodies because both are self-antigens. O group can have both because there are no self-antigens. A and B are absent. And regarding the transfusion, compatibility of the blood groups must be checked before transfusion. And always we need to check donors, antigen, recipients, antibody. Recipients, antibody. Donors, antigens must be checked. Which blood group has no antigen? That's why O can be given to any person. Universal donor. In recipient, what do we check? Antibodies. Which blood group has no antibodies? That means AB is universal recipient. That's what it is. So A group person can be given A, O, B and O. AB person can receive any blood group. AB group can be given to. O group can be given to. O group can receive. That's it. And so regarding this A and B, naturally antibodies are present. Another blood group is RH. It was first detected in rhesus monkeys, so it is given that name, and it's a protein. It is also present on the surface of RBC. A and B are carbohydrate, but this is a protein molecule. And another special feature is, for RH antigen, there are no natural antibodies in any person, whether positive or negative. By the way, 80% people are positive, remaining negative, and no person has anti-RH antibodies. Antibodies are developed by negative person upon exposure to positive. Otherwise, they are not developed. And RH grouping is very, very important in terms of the disorder. What is the disorder? Erythroblastosis. That condition can occur only when mother is negative, fetus is When mother is negative, fetus becomes positive only when father is positive. And during first pregnancy period nothing happens because fetal blood, maternal blood kept separate in the placenta. Nothing happens. But at the time of parturition or delivery, placenta ruptures when blood capillaries, maternal and fetal also rupture, and there is some amount of mixing of the blood. Okay, and due to this, when Rh antigen of the fetus enters mother circulation, by the time it circulates and it is detected by mother's immune system and start producing antibodies, the child is delivered. So first child is not, but due to this first delivery, mother becomes sensitized against Rh and there is formation of anti-Rh antibodies and memory cells in the mother. So, if the second baby, subsequent fetus also positive, then these antibodies can reach the fetus and can cause destruction of RBC, hemolysis leading to anemia, jaundice symptoms, and this is called hemolytic disorder of newborn, HDN, or erythroblastosis fetalis. Because in the fetus, due to large scale destruction of RBC, bone marrow tries to compensate by producing cells faster, releasing in immature stage itself, erythroblast. That's why the disease is called as erythroblastosis fetalis. And positive, negative, this refers to RH. Which blood group has 3 antigens? AB, positive. No antigen. O negative. 2 antigens can be seen. AB negative, B positive, A positive, considering both RH and A group. And erythroblastosis affected person always. RH negative, positive, homozygous, heterozygous, give me clarity. Huh? RH positive person only affected by erythroblastosis and it is heterozygous because the disorder occurs when mother is negative. Mother is negative, father is positive. That means the child must be definitely heterozygous. So erythroblastosis affected person always heterozygous positive. Okay. Now coming to the clotting, I just discussed the main points. Whenever there is injury, damaged platelets release some factors. Damaged platelets present in the blood release some factors. Same time, damaged tissue also releases certain factors. These factors are activated in a cascade manner. Enzymatic cascade reactions occur. Remember, this is extrinsic pathway because it's from outside tissue and this is blood, platelet. It finally forms an enzyme complex called thrombokinase. It is not a single factor. Enzyme complex. Thrombokinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. And the fibrin threads form a network in which the blood cells get trapped to form a clot or coagulum. A dark scum, jelly-like solid structure is formed. That is called clot or coagulum. With this, further bleeding is stopped. That's it. Most important point for all these reactions to occur, calcium ions are needed. Without calcium, clotting cannot occur. That's all about it. Next one. Lymph. Lymph is nothing but tissue fluid collected by the lymphatic system. Look at this. When blood flows through the blood capillaries, plasma with dissolved substances diffuses out into surrounding tissues and it is called as tissue fluid or interstitial fluid. The mineral composition of tissue fluid is same as plasma. It differs from plasma in the absence of protein. Large proteins cannot come out. Okay, so that's the difference. And formed elements do not come out except for some leukocytes, diaphysis. That's why in the book also it is given RBC platelets never come out, some leukocytes also. So it contains pure formed elements. Okay, most of them are not there. That's the reason why it is colorless because there are no RBC. Lymph can clot very slowly. Platelets, RBC do not come out. Okay, some of these inactive factors gradually become activated, we clot slowly but not normal pace. Okay, and now coming back, this tissue fluid which is formed from blood entering the tissues is the mediator between blood and or middleman between blood and tissue. Along with this fluid only, gases, nutrients are made available to tissue. Or exchange of substances between blood and tissues occur only by tissue fluid or interstitial fluid. And this tissue fluid or interstitial fluid is collected by, these are blood capillaries. Just like that, there is another network of capillaries called lymph capillaries. They collect this tissue fluid. Lymph capillaries join to form larger, they join to form lymphatic ducts, and these ducts open into major veins, supply vein. That means the tissue fluid is returned to the blood, actually derived from the blood and the lymphatic. All these together constitute lymphatic system, and this returns it again back to the blood circulation. Okay, next one also helps in transport of some nutrients and hormones also. Fats are absorbed in the intestine. Lymph vessels called as lacteal, present in the intestinal villi, into which these fats are absorbed. Then circulatory pathways. Regarding circulatory pathways, the two systems are open and closed. Blood pumped by the heart flows to the body cavities. Sinuses, open system. All tissues, organs are bathed in the blood, open system. And there is no capillary connecting all blood vessels, open system. And blood in open system is called hemolymph because tissues are bathed in the blood. And if blood flows only through a network of blood vessels, it is called as closed system. Blood never comes out of the network of capillaries. Arteries and veins are joined by capillaries, and diameter of the blood vessels vary. It never comes out. It is called closed. Which is advantageous? Closed. Because blood flow can be precisely regulated in the closed system. The two phyla showing closed, Annelida and Chordata. Other phyla exhibit open. Not all other. First phyla do not have circulatory system at all. And coming to the vertebrate circulatory pathways. Shall I ask or explain? Fishes, how many chambers are there? Two chambers, one auricle and one ventricle. And in this case, what type of blood is received? From body parts. Deoxygenated blood is received. From atria it goes to ventricle. Now ventricle pumps it to gills, where blood becomes oxygenated, and oxygenated does not go back to heart, directly supplied to the body parts. So blood flows only once in the heart in one circulation, single circulation. What type of blood is flowing through the heart? Deoxygenated. Heart receives only? Deoxygenated. Pumps only? Deoxygenated. Such a heart is called venous heart. And heart pumps the blood only to gills, branchial heart. From the gills, blood goes to all the body parts. That is single circulation in fishes. Amphibians and reptiles except crocodile, 3 chambered, 2 atria and 1 ventricle. From body parts, what type of blood? Received by? From lungs, oxygenated blood by left atria. And both the blood goes to single ventricle, where there is mixing of blood, and the mixed blood is now pumped to all the body parts. To body parts as well as to lungs. That means now you can see 2 circulations, but they are not completely separate. This is the common pathway, so it's called incomplete double circulation. Nature of blood pumped is mixed blood. It receives separately right and left, but mixed blood is pumped by the ventricle. And crocodiles, birds, mammals have completely 4 chamber, and left atrium receives from left atrium, left ventricle, left side oxygenated blood. It is supplied through systemic arteries to all the body parts, becomes deoxygenated, returned to right. What is the circulation called? Systemic circulation. Actually, this point, double circulation is discussed. I complete that part now. That means systemic circulation starts from left ventricle, ends in right ventricle. Now, right ventricle pumps the deoxygenated blood to lungs, where the blood becomes oxygenated and returned to what is this circulation? Starts in. Right way. Ends with. Done? That is complete. And there is no common point. Heart is pumping oxygenated blood separately, deoxygenated blood separately, no mixing of blood. So it is complete double circulation. Next one. Human circulatory system. Our circulatory system includes heart, blood vessels, arteries, veins, capillaries and all that. So first let us start with the description of the heart. It is mesodermal in origin, size of a clenched fist. It is located between the two lungs in a space called mediastinum in the thoracic chamber, slightly tilted towards left side. It is enveloped by double-layered pericardium with pericardial fluid which reduces the friction near the surface due to repeated contractions, regular contractions and relaxations. And four chambered. Upper chambers, smaller chambers, muscular, more or thick walled ventricles. Pumping chambers, receiving chambers, atria. And externally atria ventricles are separated by deep groove called coronary sulcus. Internally septa, externally grooves. Okay, interventricular groove separates the ventricles externally. And then to consider the structure, the two atria are internally separated by. It's a thin muscular septum and it has a depression called fossa ovalis, which is a remnant of foramen ovale of the embryonic stage. Okay, and then right atrium receives blood from superior vena cava, inferior vena cava, and coronary sulcus from the wall of the heart. Coronary sinus from the wall of the heart. So it is receiving superior vena cava, inferior vena cava from all other body parts as well as from the heart through coronary sinus. Nature of blood, deoxygenation. Left one receives oxygenated blood from lungs through how many? 4. 2 from each lung. 2 from each lung, totally 4 veins open into the left atrium. Okay, and atria, ventricles, each side separated by intera. Atrioventricular AV septum. And what is the nature? Fibrous connective. This point is important. This septum is fibrous connective tissue. That's why it acts as insulator, does not allow impulses to be conducted directly from atria to ventricle. Okay, and each of these septum, that is right and left, they have opening. Atrioventricular opening. Left atrioventricular opening is guarded by bicuspid or mitral valve. Right is guarded by. These valves allow the blood to flow from atria to ventricles and prevent the backward flow. Backward flow from ventricles to atria is prevented. 3 cusps, 2 cusps, you know that mean. And then ventricles are thick walled, highly muscular. The 2 ventricles are separated by thick muscular, thin muscular, thick muscular, fibrous connective. That is about the septa. Okay, and the wall of the ventricle is very thick and it is raised or folded into ridges called as columnae carneae. Some of them are very large and conical in shape. What are they called? Papillary muscles. AV valves are connected to papillary muscles through collagenous strands called chorda tendineae. Chorda tendineae help to keep the valves in their position, prevent them from bulging or being pushed towards the atria. Okay, that is about it. And from the ventricles arise arches. Left ventricle, aorta, which is a main blood vessel supplying to all the body parts through arteries. And from the right side arise pulmonary arch. Pulmonary arch divides in 2 branches supplying to both the lungs present on either side. And openings of pulmonary arch or aorta both are guarded by semilunar valves. or you can call pulmonary valve or aortic or systemic valve. Semilunar valve, three half moon shaped structures are there. And these semilunar valves allow the blood from ventricles into arches, prevent the backward flow. Done? That is about it. What's the next one? Nodal tissue. I show this here only. This yellow colored part. In the wall of the heart, a part of cardiac musculature is specialized to generate action potential on its own. That means auto regulation. Intrinsically, it can generate this action potential, and that specialized tissue is called as nodal tissue. This nodal tissue is spread all over the heart. It includes a patch of tissue upper right corner of right atrium called as sinoatrial node. Another one, lower left corner, it is close to AV septum, so it is called as AV node. From the AV node arise AV bundle. It pierces through the AV septum and emerges on the top of interventricular septum, and in the septum, what is this called? AV bundle. Immediately divides into two branches, right and left bundle branches. All this is called as. And at the apex, it splits to form many Purkinje fibers spreading over the walls of ventricles. Normally, all these structures or components of nodal tissue have the ability to generate, but the number of potentials that they generate per minute varies, maximum by SCA node nearly 70 to 75 times, and it generates and maintains the heart rate. Rhythmicity of the heart functioning is maintained, so it is called as pacemaker. If it fails, AV node also can generate, but the number is less, 40 to 60 times. Purkinje fibers can generate 20 to 35 times. That means all can generate, but the number of potentials vary. Is that clear? Next one. Cardiac cycle. Remember the method how to calculate duration? Duration of what? Cardiac cycle. All the events which occur in one heartbeat constitute cardiac cycle. And if heart beats 70 to 75 times per minute, how much time is taken for one cardiac cycle? 0.8, that is given data. And if they give last time was 85 times a day, whatever is the question, if you are asked to find out the duration, 60 by that number. 60 seconds, 1 minute. So 60 by that number. Suppose heart beats 100 times. What is the duration of cardiac cycle? 60 by 100, 0.6. Okay. So if heart rate increases, duration? Duration of the cycle decreases. And this is categorized into normal duration 0.8 second, and we discuss this as 3 phases. Contraction is systole, relaxation is diastole. One heartbeat includes systole and diastole. First we start with what generates? SCA node. SCA node causes, when SCA node generates action potentials, they spread over the walls of both the atria. Both the atria contract simultaneously, and now that is picked up by AV node through bundle of his Purkinje fiber spreads to the walls of, making both the ventricles undergo. Both the atria contract simultaneously. Both the ventricles contract simultaneously. Both atria ventricle contract simultaneously. Wrong. Not possible because of this fibrous. Ventricular contraction always occurs after atrial systole only, but never simultaneously. Okay. And now coming to this. So actually first step is SCA node causing atrial contraction. That means by the time atrial systole starts, heart is in which state? That means all 4 chambers are relaxed. AV valves are open. That means blood is flowing from atria to ventricle. That means ventricles are getting filled with blood, and here they are all relaxed. That's why it is called as passive ventricular filling. Ventricles are getting filled passively in relaxed condition. And semilunar valves are closed. AV valves open, semilunar valves are closed, all four chambers are relaxed. That's the situation. Now SA node generates the action potentials which spread over the walls of and both the atria contract, which is called atrial systole, lasts for about 0.1 second. When atria contract, pressure in the atria and additionally, before that also blood is flowing from atria to ventricles. Now due to the contraction, additional 30% of blood flows into the ventricle and this is called as, that's why it is active ventricular filling. That's all about contracts, additional 30% blood is pumped into the, sent into the ventricles. It is called active filling. And now, next phase, after atrial systole. When atria are contracting, meanwhile action potentials are picked by AV node and ventricles begin to contract. Now it is ventricular, lasts for about 0.2. Here, end of atrial systole, beginning of ventricular systole and end of ventricular diastole. Now ventricles start contracting means their diastole ends. It's called end diastole. Actually here we talk about these volumes also. End diastolic volume. How much? Approximately 120 ml. That means each ventricle is filled with 120 ml of blood by the end of its diastole. Okay. And this is the beginning of atrial diastole. Ventricular systole coincides with atrial diastole. And when ventricles contract, pressure in the ventricle due to which blood attempts to go back and that is prevented by, that means AV valves with further increase in the pressure, semilunar valves are, blood rushes from ventricles into arteries and closure of AV valves produce the first hearts of lung. And then ventricle systole ends. At the end of the ventricular systole, still some amount of blood is present which is called end systolic volume approximately. That means how much is pumped out? That is called as stroke volume. How do you define? Volume of blood pumped by each ventricle in one heartbeat is called as or beat volume. Beat volume or stroke volume. Okay. When you calculate that for 1 minute, cardiac output. Pumped by each ventricle per 1 minute is cardiac output. So cardiac output is equal to SV into heart rate. So if heart rate increases or stroke volume increases, automatically cardiac output increases. Okay, next one. End of ventricular systole, is it the end of atrial diastole? No. Now ventricles begin to relax, atria already relaxed condition, that's why it is called joint diastole. It is 0.4 seconds. Now pressure in the ventricle with pressure decreasing the ventricle, blood from the arteries tries to come back, which causes semilunar valves to close. This produces the second heart sound. With further decrease in the pressure, meanwhile pressure in the atria increases because of blood received from all body parts, and this increased pressure in atria forces open the AV valves and blood starts flowing into the, so AV valves open and filling of the ventricles begin, which is passive because all chambers are relaxed. That means we have come back to the first condition. That's the cycle. Atrial systole duration 0.1. Atrial diastole 0.7. Ventricular systole, ventricular diastole 0.5. Joint diastole, joint systole not because it is not possible. Okay, next one. Active filling, ventricular filling. Ventricular ejection occurs during systole. Ejection means sending out the blood. Ventricular systole. So this is about all we discussed. Heart sounds are of clinical importance. Now comes ECG. Electrocardiogram. Machine is graph. The record is gram. And technique is graphic. And Standard ECG. It is nothing but recording of electrical activity, and it is done by connecting the machine to the body at three specific locations, both the wrist, left hand. This is standard ECG. If a detailed analysis is required, multiple leads, about 12 at different positions of the chest also connected. And here we are discussing only standard ECG. And this is represented in the form of graph. I mean, this wave pattern can be seen. PQRST. P represents atrial. Meaning depolarization is not contraction. Depolarization results in contraction. P is atrial depolarization, meaning atria become electrically excited, meaning literally action potentials generated by SCA node are spreading over the atria, stimulating them. That is happening during. Shortly after that, atrial contraction occurs somewhere here. Okay. And then QRS complex. This is ventricular. That means spread of the action potentials in the walls of ventricles. Our ventricles are becoming electrically excited, and this is represented by QRS. And shortly after the Q, actual contraction begins, and it is T wave represents repol. That means from excited state coming back to ventricular repolarization. That means repolarization is followed by relaxation. That means relaxation or diastole occurs after T. End of T is end of two systems. Ventricular. If nothing is mentioned, take it as ventricle, because in heart main pump. Heart is a pumping organ. Which ones are pumping chambers? They are the important ones. So ventricular systole starts after Q and ends with T. And remember, first heart sound love occurs during ventricular. That to beginning of ventricular systole. First step is closure. That means it occurs somewhere here. Or not Q. Contraction starts after Q. Don't take Q. Contraction starts after Q. That means it may correspond to R or S. Okay. And second sound occurs during joint diastole. After T. If you relate the sounds with this. And number of QRS complexes indicates the heart rate. Which wave is not represented here? Wave representing atrial repolarization. Atrial depolarization is there. Ventricular depolarization, ventricular repolarization. Atrial repolarization, there is no separate wave because it occurs around the same time when QRS happens. And QRS occurs with high amplitude, it masks the atrial repolarization. Okay, and this is a standard ECG, and any person ECG normally looks like this. If it shows any variation, it indicates some problem with the heart functioning. So it helps in clinical. Next one. Double circulation I discussed. Here you need to learn about those blood vessels. Blood vessels, arteries, veins, all these are there. Arteries, veins, walls are made up of three layers. Outer, tunica externa, made up of fibrous connective tissue rich in collagen. Middle, tunica media, made up of smooth muscles and elastic fibers. Inner, tunica interna or endothelium, simple squamous epithelium. Both arteries and veins have these three layers. Difference is tunica media is thin in veins, thin in, very thick in. That means more elastic fibers are vessels are arteries. Which ones are having more wider lumen? Veins. Narrow lumen. Thick walled. Thin walled. Valvular. Deeply located. Superficially located. Which blood vessels always take the blood away from the heart? Towards the heart. Supplying to body parts. Collecting from body parts. Which one usually carry oxygenated blood? Except. Deoxygenated. Except. Okay, okay. And which vessels retain the shape even after blood is removed? Arteries, because they are thick wall. Veins collapse because they are thin wall structures. And coming to the special points. Normally veins carry the blood towards the heart, but there are some veins after collecting blood, instead of carrying to heart, they carry it to some other organ. We call them as portal. Which portal system we have? Hepatic portal system, from intestine or digestive system to liver. Present in frog? All vertebrates have hepatic portal system, no exception. We don't have. It is present in reptiles, amphibians, mammals and cyclostomes do not have. Okay, and we also have coronary circulation. Aorta supplies the blood through coronary arteries to myocardium of the heart and collected by 4 coronary veins which open into coronary sinus that opens into right atrium. So there is supply of blood to the heart musculature and again collecting it back. Okay, coronary musculature. And here points are which blood vessel has highest amount of urea? Hepatic, not portal, hepatic vein, because hepatic vein collects from liver. Liver is a place where ornithine cycle, urea synthesis occurs. So blood collected from liver is rich in urea. Which blood vessel has highest amount of nutrients? Hepatic portal vein, because it collects from intestine where food is digested and absorbed. Okay, and least amount of urea, because kidney filters out the urea, blood coming out of kidney would be having less amount of urea. So few related points. Double circulation I discussed already. What is the next one? One important point. See, this is a capillary. If you observe, see, vein is having thin wall and lumen is wide. This is thick wall, lumen is narrow. Capillaries, just inner endothelium is present. So what is the site, actual site where exchange of substance? Capillaries. Exchange of substance never occur arteries and veins or arterioles and venules, the branches, no, happens only at capillaries because there's only one lined by single squamous epithelium. Okay, next one, regulation of the cardiac activity. Normally there is no need to alter the heart function. Heart controls itself, intrinsically it regulates itself because of the nodal tissue. Nodal tissue takes care of. So, heart contracts, works on its own without any external stimulus normally because it is intrinsically regulated. But whenever there is a need for alteration, like a person starts running or doing some exercise, now there is a need for increase in the heart rate. So, whenever alteration is needed, neural and endocrine systems help in regulating that. First, neural one. In the medulla, there is a center. Cardiovascular center located in medulla. This center sends signals to heart via autonomic nervous system. Autonomic nervous system includes 2 components, sympathetic and parasympathetic. Sympathetic signals increase the heart rate, strength of ventricular contraction, cardiac output. Whereas parasympathetic signals decrease the heart rate, slow down the speed of conduction and decrease the cardiac. So both are antagonistic. Depending on the need, they carry the signals. In addition to that, endocrine, adrenal medulla secretes epinephrine and fight and flight hormones. They increase heart rate. Thyroxine also increases because it increases BMR, automatically increases the. Next one, disorders. Normal blood pressure, 1, systolic pressure, diastolic pressure, and this is a normal pressure, pressure exerted by the blood flowing in the blood vessels. Instrument used, sphygmomanometer. And when repeated checks reveal high values, what is the limit? 140 by 90, then it is called as high blood pressure or hypertension. Hypertension can cause damage to heart, brain, kidneys, etc. So it should be under control. Next one, coronary CAD or atherosclerosis. It is nothing but deposition of calcium, cholesterol, fats, fibrous tissue on the wall of the coronary artery. This is called plaque. Deposits called as plaque. Due to plaque formation, lumen of coronary artery becomes narrow. When lumen becomes narrow, blood supply to the heart wall decreases. Clear? That is the CAD disease. And due to this decreased blood flow, oxygen supply to cardiac musculature, which can cause acute chest pain, condition called as angina pectoris. Angina pectoris is due to reduced oxygen, that is blood supply, which can be caused by CAD also. Narrowing of the lumen, and it can occur any age group, common in elderly people. Then 3 points: heart failure, heart attack, cardiac arrest. What? First one, heart is pumping the blood but not efficiently to meet the demand of the body. That condition is called as sudden damage of the myocardium or localized death or necrosis of the any of the words, correct. It is called heart attack or myocardial infarction. Third one, cardiac arrest. Heart simply stops beating. It may be problem in the nodal tissue, not generating action potentials. Okay. So sudden stoppage is called as cardiac arrest. Next one, excretory system. What is the need for excretion? Actually, in our body, substances tend to accumulate due to excess of ingestion or formed due to metabolic process. They include different electrolytes, K plus, all those, calcium and all that, or nitrogenous waste materials, any substance. These must be removed partially or completely depending on their nature. Okay, and here in this talk, the process of elimination of waste materials is called as excretion, and here we are discussing, focusing mainly on nitrogenous waste, ammonia, urea and uric acid. Highly toxic? Least toxic? Urea always in between only. So I am asking top and least. Okay. Which one helps in highest water conservation? Huh? I asked which one helps in water conservation? Uric acid. Which one needs more water expenditure? Water conservation is not possible. Ammonia, because it needs lot of water. Which one does not need energy expenditure? Ammonia. More energy expenditure is needed for, because conversion of ammonia to these substances needs more energy. So energetically more expensive is uricotalism. Same point. Uricotalism. Water conservation highest, uricotalism. Water expenditure highest, least toxic. Which substance can be eliminated in the form of paste or pellet? Uric acid. Actually, terrestrial animals needed to eliminate less toxic substances like urea, uric acid, because they cannot afford to use water for excretory purpose. So, elimination of ammonia is ammonotalism, and it is observed in bony fishes, aquatic insects, aquatic amphibians. And regarding ammonotalism, one point is there. Fishes, just now bony fishes, they have kidneys, but kidneys do not play significant role, because ammonium, which is highly soluble, readily dissolves in water and eliminated as ammonium ions through their body gill surface. Lower animals body surface. All lower animals also excrete ammonia, completely aquatic. These eliminate Through gill surface in the form of ammonium ions. That's why kidneys play less significant role there. And then ureotelism. In ureotelism, ammonia is converted to urea by ornithine cycle which occurs in, so urea synthesis occurs in, urea elimination occurs kidneys, and urea is less toxic, it needs less water for its elimination. As it is less toxic, some amount of urea is retained in our medullary interstitium to maintain high or desired osmolarity. Last one, examples: mammals, terrestrial amphibians, marine fishes, cartilaginous, contact fishes. And ureotelism: birds, reptiles, insects, land snails. And here water is not needed, it is not soluble also, literally insoluble. Eliminate as paste or pellet, helps in lot of conservation of water. Next one, excretory organs. First time special excretory structures appeared. Platyhelminths, platforms, flame cells. Flame cells primary concern with or you can say ionic and fluid volume maintenance. Okay, and flame cells are seen only in flatworms? No. Flatworms, rotifers, some annelids, cephalochordates. What is example? Amphioxus or branchiostoma. They all have these flame cells or protonephridia. Next, Ascalmenthes. Excretory canals are present. Excretory tubes are canal. Some possess renity gland. Renity gland occur only in some. Main excretory canals are tubes. Annelida. Nephridia. Arthropoda. Malpighian tubules. Coxal glands. Green glands or antennal glands found in crustacea. Prawns given here also. Okay, then so there are different structures. Mollusca, metanephridia or kidneys are present. Echinodermata, no special excretory system is there. Hemichordata, proboscis gland is there. And among chordates, urochordates have neural gland. Cephalochordates have protonephridia. All vertebrates have kidneys. None? Okay, next one. Next. Our excretory. Our excretory system includes a pair of kidneys, ureters, urinary bladder and urethra. These are paid and these are unpaid structures. And kidneys are metanephric type, advanced condition. And what is this blood vessel? Dorsal aorta supplying blood through renal arteries to kidneys, and blood is collected by renal veins which open into inferior vena from lower parts, inferior vena cava. And what's the shape of the kidney? Kidneys are located dorsally on either side of backbone at the level of last thoracic, third lumbar. Trachea bifurcates at on 12th vertebra. Made mistake and now remember very well. Okay. So, okay. And that is the position. And one more thing. Kidneys are described as retroperitoneal because dorsal side of kidney directly attached to body wall. Only ventral side of kidney is lined by peritoneum. That means kidney is behind the peritoneum. So, they are called retroperitoneal organs. Okay. And shape of the kidney? Outer surface is convex. Inner surface is concave. Middle of the inner concave there is notch. Hilum or hilus through which blood vessels, nerves and ureters pass. Whole kidney is covered by renal capsule. Dense fibrous connective tissue. Length 10 to 12, 5 to 7. 2 to 3. Okay. And coming to the other points, internal structure, anatomic internal structure. This is outer convex surface, inner concave surface, and this is ureter which comes out, and just inner to the hilum there is a funnel shaped cavity called renal pelvis. It forms cup like projections called calyces. Inside the kidney, we differentiate two regions, outer cortex, inner medulla. Medulla is arranged in the form of conical pyramids. Tips of the pyramids are called papilla. They are projected into calyces, and outer cortex extends in between the pyramids as columns of Botany or renal columns. Renal columns are columns of Botany. That's the structure. Okay. Renal pelvis, funnel shaped cavity continues as ureter, which emerges out through hilum. Done. And inside the kidney, there are the functional units, nephrons. Each kidney has how many? 1 million. Where are they located? Cortex or medulla? Both. They extend from cortex to medulla. So, okay, that part is done. Next, nephron structure. Nephron is made up of two components, glomerulus and renal tubule. Renal tubule starts with double-walled cup. In the Bowman's cup is enclosed glomerulus. Glomerulus and Bowman's capsule together form Malpighian body or renal corpuscle. And what is this glomerulus? Bunch of blood capillaries which receives the blood through efferent arteriole. One of the branches of renal artery enters, divides to form a bunch, and all these capillaries join to form efferent arteriole, which takes the blood away from the glomerulus towards the glomerulus, away from the glomerulus. Which one has more diameter? That is important, helps in filtration. So efferent arteriole less diameter, efferent has more diameter. And Bowman's capsule, double walled, lined by simple filtration site, and it is followed by highly coiled, lined by simple cuboidal with brush border, followed by loop of descending limb and thin segment of ascending lined by squamous epithelium, thick portion lined by cuboid, then DCT. Cuboidal epithelium, but brush board is not clearly seen, and DCT opens into collecting duct. And in this part, most important thing is which components are located in the cortex. Bowman's capsule, PCT, DCT. They are found in the cortex. Loop of Henle, medulla. Collecting duct extends both cortex and so vasa recta is also in. Because loop of Henle is in. And let us complete this. Efferent arteriole after emerging out divides to form a capillary network which is called as peritubular capillary network. One of the branches of that runs close to loop of Henle called as vasa recta. All these take the blood away through venules and veins from the kidney. Okay. So that is the structure. Which vessel starts with capillaries, ends with capillaries? Efferent. Normally, arterioles end with capillary. Venules begin with capillaries. Here, efferent starts with glomerular capillaries and then again divides to form peritubular capillaries. So, starts with capillaries and ends with capillaries. Done? Next one. What's the next one? Urine formation. First step, glomerular filtration or ultrafiltration. Some of the points. What type of epithelium forms the Bowman's lining? Squamous. Double layered. Outer layer cells are normal squamous cells only, but inner layer cells are specialized podocytes. They form pedicels. So they are intricately arranged, leaving fine gaps called filtration slits or slit pores. Not only that, the endothelium of this capillary also has simple squamous epithelium, and these cells form fenestrations. Endothelium forms fenestrations. Podocytes of Bowman's cup form filtration slits. Both increase the efficiency of filtration. And blood present in the glomerular capillary, fluid of Bowman's cup separated by how many layers? Same, 3 layers. It's called filtration membrane because blood gets filtered from capillaries into Bowman's cup. It's a 3-layered one. What are the layers? Endothelium, basement substance, Bowman's cell. Epithelium or the Bowman's capsule. How many cellular layers? 2. Same as that membrane only. Okay. And filtration occurs due to glomerular blood pressure. Glomerular capillary blood pressure. NCRT point. Filtration occurs due to the blood pressure of the glomerular capillaries. We talked about 3. Blood pressure, osmotic pressure, and that is called osmotic pressure, and hydrostatic pressure of the renal fluid. What are the pressures that are opposing? Blood pressure of the capillaries is opposed by colloidal osmotic pressure of the blood and hydrostatic pressure of the glomerular fluid. So these two, one direction. If you remember, this is 60. 60 opposed by and this is 18. 32 and 18. Total 50. 32 mm of Hg osmotic pressure. This is 18 mm of Hg. That means overall 50. So total this pressure is less than hydrostatic pressure. So net filtration pressure is 10 mm of Hg, which cause ultimately this high pressure is only causing the filtration. That's why in NCRT they mentioned only this pressure. Glomerular capillary blood pressure causes the filtration. Here the important points is the filtration active or passive? Passive. Filtration occurs due to pressure difference. There is no expenditure of energy. It's a passive process. Selective or non-selective? Non-selective. Because during filtration, all components of plasma with dissolved substances except the proteins get filtered. That's why such a fine filtration is called as ultrafiltration. And how much blood is subjected to filtration? 1100 to 1200 per minute, which is equal to 1/5 of the cardiac output. Cardiac output also per minute. So 1/5 of that. When this much of blood gets filtered, how much of filtrate is formed? 125 ml per minute. This is called G glomerular filtration rate. Clear? Don't get confused between these two values. This is the volume of blood that is subjected to filtration. This is the amount of filtrate. How much plasma is filtered? 55% of 1100 to 12. If at all they ask. Usually they ask the blood only. Okay, this is about the filtration. And if 125 ml is formed per 1 minute per day, how much is formed? 180. But a person eliminates. That means all the filtered fluid is not eliminated. Now in the renal tubule, filtrate is subjected to two processes, reabsorption and tubular secretion. Before going to that, one more point. Again, we discuss it anyway, it's there. JGA, juxtaglomerular apparatus. We talked about GFR. That filtration rate is auto-regulated, intrinsically regulated by the kidney itself by JGA. JGA is a sensitive region formed at the point of contact between efferent arteriole and DCT. Where DCT and efferent arteriole come in contact, cells in that region are modified. Modified smooth muscle cells in the wall of efferent arteriole. Crowded or densely packed cuboidal cells in the wall of DCT, macula densa. So macula densa and JG cells together constitute juxtaglomerular apparatus. whenever GFR falls down, JG cells produce renin, which helps to bring it back to normal. Thus, it is intrinsically regulated. Now, coming to this process, both the processes are selective. When filtrate passes through renal tubule, useful substances get reabsorbed at various regions, actively and passively. Which ones are absorbed actively? Sodium, all the nutrients, glucose, amino acids, etc., they are absorbed actively. Water is absorbed passively in the initial parts, not everywhere. Initial segments, what is absorbed passively? Nitrogenous waste absorbed passively. Nitrogenous waste passively. Urea absorption occurs in collecting duct passively. Water absorbed passively in the initial segments. Nutrients actively. Sodium also actively. This is about reabsorption. Secretion. Usually K plus, H plus, ammonia secreted into the tubule and this plays important role in maintaining ionic balance, acid-base balance or pH. Because K plus, H plus, all this contribute to the, H plus contributes to acidity, you know that. So helps in maintaining that balance. So it is also equally important. Now the next part is formation of. Actually, this function of the tubules, you don't have to learn or look at this theory also. You can simply refer to the diagram. Everything is shown. You need to make one or two changes in that, so that everything is at one place. First part, PCT. PCT is lined by simple bordered brush border. Increase the surface area, very much suited for reabsorption. And in this region, 70 to 80% of water and electrolytes are absorbed. 70 to 80%. So major site for reabsorption is PCT. And in addition to that, all the nutrients, glucose, amino acids absorbed active, completely absorbed in the PCT. Nutrients completely reabsorbed in the PCT. And in addition to that, K plus and bicarbonate ions also absorbed. You know that arrow directed outwards means reabsorption. Into the tubule, tubular secretion. H plus ammonia secreted into the tubule. That means it helps in reabsorption as well as plays a role in ionic balance or acid-base balance by secretion of these ions. Done? That's all about PCT. And then descending limb. Loop of Henle has main role in maintaining high osmolarity of medullary interstitium. Minimum reabsorption occurs in the ascending limb. See, descending limb is permeable to, nearly impermeable to. So while passing through that, only water gets reabsorbed. Due to water reabsorption, concentration of the filtrate progressively increases and reaches highest here. Done? That's it. And this absorption in PCT and descending limb is called obligatory absorption, because irrespective of needs of the body, this proportion of water absorption always occurs. Obligatory or mandatory, compulsory. And ascending limb. Ascending limb is impermeable to. It has thin segment and impermeable. It allows transport of sodium chloride passively and actively. In the book it is given actively or passively. But if they focus on that, thin segment passive, thick segment active. So actively, passively sodium chloride absorption occurs in ascending limb. No water absorption. That means renal fluid is losing the electrolytes, solids. So progressively it becomes diluted, more concentrated, more diluted in ascending. And now DCT. In the DCT, again, see, water and sodium chloride absorption occurs here also, but it is conditional absorption depending on the need of the body. Water absorption occurs by the influence of ADH, sodium by aldosterone. Okay, and these hormones are produced depending on the need of the body. That's why it is called conditional absorption. Bicarbonate ions also absorb, and K plus, H plus, if you add ammonia here, then the theoretical part would be complete. H plus K plus ammonia secreted into the tubule. In mentioned in theory, not in the diagram. So you just add it. So every time, whenever you want to revise it, you just can check the diagram. No need of going to theory again. Is that clear? And after DCT, what's the next part? Collecting duct. Collecting duct also conditional absorption of water and sodium chloride occurs. Same conditional absorption like DCT. And in this area, some amount of urea is transported into the medullary interstitium. And just add one more thing. K plus and H plus are secreted. K plus H plus secreted in the tubule. These are the changes occurring in the collecting duct. And one more thing. Collecting duct is running from cortex into the medulla. And as we progress towards inner side, osmolarity goes on increasing. As a result, it's very easy for large amounts of water to get reabsorbed, which helps to increase the concentration of urine in the collecting duct. That's it. Function of the tubules. What is the next one? Mechanism of concentration of urine. I just mentioned that and you can take the break. Okay. 2 points. Cortex 300 osmolarity gradually increases and becomes 1200. Recently you jalak outer region of okay. So basically there is ascending osmolarity from cortex to inner medulla, and here counterflow. Flow of renal fluid, the two limbs of loop of Henle is in opposite direction, counterflow of renal fluid. This is not here, it is actually located here only, vasa recta, and the two limbs, descending limb and ascending limb of vasa recta, blood flow is in opposite direction called counterflow, and vasa recta is in close proximity to. All this helps in efficient exchange of substances. And this inner high osmolarity medullary interstitium is mainly due to two substances, NaCl and urea. Now look at this. First thing, NaCl transported from ascending limb of loop of Henle is exchanged with descending limb of vasa recta and is returned again from ascending limb into medullary interstitium. Thus, always some amount of NaCl would be there in the medullary interstitium, which contributes to osmolarity. So solids, osmolarity. And second one, urea. Urea from the medullary interstitium enters into thin segment of ascending limb of loop of Henle and is again returned from collecting duct into medullary interstitium. It always happens continuously. So at any time some amount of urea would be there in the medullary interstitium, which again contributes to high osmolarity. Thus NaCl and urea contribute to high osmolarity of the medullary interstitium. And due to this high osmolarity only, water can be reabsorbed effectively, increasing the concentration of urine. Initially 300 milliosmoles, initial urine. Final urine is 1200. It is 1200. That means it is 4 times more concentrated than the initial. Okay? Okay, that is about countercurrent mechanism. Then the rest of the topic is actually easy. Remember the regulation. Kidney functioning is regulated by hormones produced by hypothalamus, JGA and heart. Our body has osmoreceptors in the hypothalamus. They are sensitive to changes in the osmolarity. Whenever there is change in the body fluid volume or ionic volume, blood volume, fluid volume, the changes are detected by osmoreceptors. Osmoreceptors stimulate the hypothalamus to secrete ADH. First thing, listen, that's enough of relaxation. When there is severe dehydration, water loss from the body, osmolarity in the body Water loss, that is concentration in osmolarity increases. That causes stimulation of osmoreceptors. Osmoreceptors are activated and they stimulate the hypothalamus to release through neurohypophysis. Synthesized in hypothalamus, releases in neurohypophysis. Antidiuretic hormone. It causes reabsorption of water in the distal parts of the renal tubule, thereby again maintains or brings the osmolarity back to normal. In addition to that, ADH also has vasoconstrictory effect. By vasoconstriction, it raises the blood pressure so that it can increase GFR also. When blood pressure increases, GFR also increases. And one more thing, when we have lots of water in the body, drinking more water, osmolarity of the body fluid decreases and osmoreceptors will be now switched off and ADH secretion is inhibited. Water is not reabsorbed, the DCT collecting duct, and that water not reabsorbed goes out along with the urine. Urine becomes diluted. Urine becomes dilute in the absence of, that means when it is present, it makes the urine or prevents dilution of. That's why it is given the name antidiuretic hormone. Done? That is one regulatory mechanism. Second regulatory mechanism, it is from JGA. Whenever there is fall in GFR or glomerular blood flow or glomerular blood pressure, this stimulates JJ cells to release. Renin acts on angiotensinogen into angiotensin 1. 1 is converted into angiotensin 2 by angiotensin converting enzyme produced in the lung, pulmonary capillaries. And this angiotensin 2, it is a powerful vasoconstrictor, raises the blood pressure, which in turn raises the GFR. And it also acts on adrenal cortex, stimulate the production of aldosterone, which causes reabsorption of sodium and water, and with water reabsorption, blood volume increases, blood pressure, and when blood pressure increases, automatically GFR also increases. Taken care of. And this mechanism is called ROS, renin, angiotensin, aldosterone, RAA system, ROS mechanism or ROS system. And what's the third one? Heart. When more volume of blood reaches the heart, more volume means more pressure also, atrial wall would be stretched. Because more wall, receiving chambers are atria. So more blood received, atria wall stretches and it stimulates the wall of atria to secrete AN, atrial natriuretic peptide or factor. What does it cause? Vasodilation. By vasodilation, blood pressure decreases. ROS mechanism increases, this decreases. So it's a counter check to the ROS mechanism. Not only that, this hormone also inhibits absorption of sodium and water. That's why sodium not reabsorbed means appears in the urine. That's why it is called natriuretic, presence of natrium, sodium in the urine. So that's all about regulation. What's the next one? Micturition. Urine formed in the kidneys is transported by ureters and remains stored in the, it remains stored till it receives a voluntary signal from CNS, and it involves the process of expulsion of urine is called as micturition. Micturition, and it involves micturition reflex. What happens? When bladder is full, wall of the bladder is subjected to stretching. When bladder wall stretches, stretch receptors in the wall are stimulated. They send the signals to CNS. What type of signal? Sensory or motor? Towards CNS, sensory. And now CNS sends motor signals to the bladder, which causes two functions. Contraction of bladder muscles, simultaneously relaxation of urethral sphincter. Urethral sphincter regulates the opening and closure of urethral aperture. So when when it relaxes, it opens and urine is expelled or passed out. Okay, contraction of bladder muscles and relaxation of urethral sphincter, both are caused by that. This mechanism is called as micturition reflex. Okay, next, gentle points. Pale yellow color, urochrome, slightly acidic with a pH of 6. Amount of urine eliminate per day 1 to 1.5. Amount of urea eliminate per day 25 to 30. Urea, urine, both are different. And analysis of urine also helps in diagnosing some diseases like diabetes, mellitus, glycosuria and ketonuria. So that is information there. And then other organs, just points. Lungs eliminate carbon dioxide 200 ml per and water in the form of water vapor. Next, liver releases bile juice into the intestine which contains bile pigments, bilirubin and cholesterol, degraded steroid hormones, some vitamins and drugs. Sweat glands of the skin. Primary function is cooling effect, maintaining body temperature, but when it produces sweat, along with that, sodium chloride, urea, lactic acid. NaCl, urea, lactic acid along with the sweat eliminated. Sebaceous glands secrete oily substance sebum. It contains hydrocarbons, waxes, sterols. Saliva also contains some amount of urea. Done. Next one. Disorders. Accumulation of urea in the body fluids is called as uremia, and if urea levels increase, automatically more load is there on the kidney and it can lead to kidney damage. And what is a boon for these patients? Hemodialysis. That is, blood is subjected to dialysis process, and dialyzing unit, also called artificial kidney, what does it contain? It contains cellophane tubes. Cellophane tube is permeable to substances, and they use a dialyzing fluid in this machine. Dialyzing fluid has same composition as plasma except for nitrogen. So dialysate has no nitrogenous base. All other components are present. Okay, and in this process, from the patient, blood is collected through artery and add heparin to prevent clotting and allow the blood to flow through that. Blood is rich in nitrogen. Person is suffering from uremia, nitrogenous base. Surrounding dialysing fluid has no nitrogen. So there is a concentration gradient from blood to dialysing fluid. Along the gradient, not against, along the gradient, those nitrogenous base, they come out of the blood into surrounding dialysing fluid. So by the time blood comes out of this unit, it is cleared of most of the nitrogenous base, which is then returned through vein after adding antiheparin. Okay. Next one, renal calculi. Formation of insoluble masses like oxalates, also called kidney stones. Inflammation of glomerulus of nephron is called glomerulonephritis. And if the kidney problem is severe, what is the solution? Transplantation. Same gentle point. Before usually taken from close relative, blood group testing and tissue matching must be done. And after the transplantation, needs to use immunosuppressors throughout the life. Next one. Locomotion. All living organisms exhibit movements. Simplest form of movement is amoeboid movement by streaming movement. Other than the cilia, flagella, muscles, all these cause movements. If change in the body part involves change in the location or place, it is called locomotion. All locomotions are, but all movements are structures which help in locomotion need not be different from other structures causing other movements. Two examples are given. In paramecia, cilia help in locomotion. Same structure also help in collecting the food through cytopharynx. Hydra, tentacle, help in locomotion as well as capturing the food. So same structure which is helping locomotion can also help in other kinds of movements in the body. And then muscles also, changes in the body posture, locomotion, muscles, bending, straightening, all this change in the body pose. Same muscles are used. And then purpose of locomotion. Animals move from place to place in search of food, space, shelter, to escape from predators, dangers, favorable conditions, or breeding purpose, or to reach the mate, different purposes. And the type of locomotion depends upon habitat and demand of the that is the introduction part. Now coming to the other points. In human body, three types of movements are seen. Amoeboid movement exhibited by leukocytes of the blood, macrophages of tissues, and they produce pseudopodia. Amoeboid movement is due to pseudopodia. Pseudopodia formation occurs by streaming of protoplasm, and cytoskeletal structures like microfilaments also help in pseudopodia formation. Okay, ciliary movement. Cilia form lining for hollow organs. Already you have learned bronchioles, fallopian tubes, etc. And cilia cause movement in a specific direction. Fallopian tubes transport of ova. Trachea, dust particles, specifically towards the pharynx. There are so movement of these come under that. Then flagellar movement not in our body. Flagellar movement exhibit protists like euglena and lower animals like sponges, where coerocytes, flagellar cells cause movement of water through the water canal system. And in all higher animals, flagellar movement is exhibited by tail of the spermatozoa. That's it. Both the cilia and flagellar outgrowths of the cell membrane. Now we focus on the muscle. In higher animals, most of the movements are caused by muscles, especially locomotory movements. Muscular system, skeletal system, neural system work in coordination to cause the movements. And types of muscles. What is the body, percentage of muscle tissue in our body? 40 to 50%. Number of muscles? 60%. And muscles are capable of excitability, contractility, extensibility, elasticity. In response to stimulus, they contract, shorten, relax, lengthen, shorten, lengthen, and are responsible for various movements. These are classified based on location, appearance, nature. Based on the location, three types. Attached to skeletal structures, skeletal muscles, walls of visceral organs, visceral muscles, wall of the heart, cardiac muscles. Based on location, three. Based on appearance, two. Striated and based on the nature, voluntary. Skeletal muscles are described. Striated, voluntary. Smooth muscles. Cardiac muscles. Which muscle tissue forms branching network? Also characterized by intercalated disc, elongated cylindrical, skeletal muscle, skeletal, fusiform, short branch, cardiac. And you know the statements. All voluntary are striated, but all striated are not voluntary. All smooth muscles are involuntary, but all involuntary are not smooth, exception cardiac. So like that statements can be asked from that part. And regarding example, skeletal muscles, locomotion, changes in the body posture mainly by skeletal muscles. Pumping of the blood, cardiac. Transport of substance in the body through hollow organs, smooth. Example, ova movement in the fallopian tube, muscle. Food transport in the stomach and intestine, because intestine has smooth muscles. So examples are given there. Next one, skeletal muscle. Skeletal muscles are arranged in the form of bundles. Bundles are called as fascicles. All these are the bundles. All the fascicles are held together by collagenous tissue called as fascia. Collagenous tissue called as fascia. And anatomical unit, muscle fiber. So literally it is equal to cell, and this muscle fiber elongated and cylindrical, covered by sarcolemma, sarcoplasm. It is multinucleated. Remaining two are uninucleated. Skeletal muscles are multinucleated, syncytial, and nucleus is peripherally located, not central, peripheral located. This multinucleated condition includes the muscle fiber is formed by fusion of many muscle cells in the embryonic stage. And endoplasmic reticulum is called. And it's a reservoir of calcium. Mitochondria are called sarcosomes. Functional unit is sarcosome is mitochondria. And in the sarcoplasm, you can see a number of parallelly arranged fibrils. And these fibrils are in turn made up of myofilaments. Two types of filaments, thin or active, thick or myosin. And smooth muscles do not have this thin and thick. All muscles have actin and myosin. But in striated muscles, they are arranged regularly, alternate manner, thin, thick, which gives striated appearance. They are not arranged in such a regular fashion. That's why they are non-striated or smooth muscles. And now we need to discuss the structure of myofibril. Look at this diagram. This is part of the myofibril. This is dark band, light band, again dark, light. So alternately dark light bands are present. They are called I band and A band. Isotropic, I band, light band. Dark one is anisotropic. And I band is, why it appears lighter? Because it has thin filaments. A band has thick filaments, so it appears darker. Center of the I band, there is elastic fiber called as Z-line or cross-base membrane. And all what filaments are there in the eye? Thin. All thin filaments are attached to Z-line. And the region between two successive Z-lines is called sarcomere functional unit. That means one sarcomere includes one complete A band and two halves of I band on either side. Remember all that part? And now coming to the description, the same thing is shown here. This is wherever thick filaments are there, that part is A band, and the region where only thin filaments are present is I band. So again this is I band. I band is bisected by Z line. All thin filaments of I band are attached to Z line. And center of A band, all thick filaments are held together by a fibrous M line. What is this called? M line. Thin filaments of I band, they extend into the sides of A band, partially overlapping the thick filaments, partially, not completely, so that the central portion of A band has only thick filaments, not overlapped by thick filaments. So that part appears lighter than the side regions and is called H zone or tensile space. Portion where thick filaments not overlapped by thin filament called as H zone, and M is a central fibrous layer. All thick filaments are held together. That is the structure. Okay. Next one. Structure of contractile proteins. Thin filaments also called as actin, but it doesn't mean that thin filament is made up of only actin. Thin filaments, they are made up of three proteins: actin, tropomyosin and troponin. But major one is actin, so they are simply called as actin filaments. And look at this. This is one filament, this is the other one. That means there are two filaments of actin. They are called F-actins. How many are there? Two. Helically bound around each other. And each F-actin is a polymer of many monomers called as G-actins. And these are having active sites for to bind with the myosin. And running along with these F-actins, there are two filaments of tropomyosin running along the F-actins, and tropomyosin also actually masks the active site, not mentioned in the book, but it along with the troponin plays important role. And arranged at regular intervals on tropomyosin, you can see complex for troponin made up of The one which binds to tropomyosin is TNT. Calcium binding site TNC, which inhibits interaction with an actin myosin TNI. And as given in the NCRT, in a resting muscle, one of the subunits of troponin masks the active site. So that is about it. And actin myosin are main contractile protein. Troponin, tropomyosin, they play important role in masking and unmasking. So they are called regulatory proteins. Done. Next one, thick filament made up of many monomers. Monomers are called meromyosin, and each one has head with short arm. This is a short arm, globular head, rounded head, short arm, and the tail portion. Okay, helically around, wound around each other. And head and short arm made up of heavy meromyosin. Tail is made up of light meromyosin. Head is having two binding sites, one for actin and one for ATP. Head itself is a site of ATPase, which can break down that ATP molecule. Okay, and these myosin molecules, they are arranged in such a manner that head with the short arm is raised, projected outwards, making an angle with the filament, and that is called as cross arm. Called as cross arm. Next one, contraction mechanism. Actually, here you need to learn that definition. What is motor unit? Muscle contracts only when it receives motor impulse from the CNS. One motor neuron, all the muscle fibers innervated by that together constitute one motor unit. And when you consider that motor neuron, these are the axon terminals. Axon terminals are provided with synaptic knobs, and this is sarcolemma. So this junction between neuron and the muscle cell is called neuromuscular junction or motor end plate. Now let us discuss the mechanism. Which theory explains it? Sliding filament. Why is that theory given that name? Because contraction occurs due to sliding of thin filaments over the thick filament. And first step, motor impulse reaches the neuromuscular junction. Once it reaches, the synaptic vesicles release neurotransmitter into the neuromuscular junction. Sarcolemma has receptors for. Once neurotransmitter binds to that, actually myasthenia gravis, this is the problem. Receptors are blocked. So cannot bind, cannot stimulate the muscle. So normally it binds to that and stimulates the sarcolemma. Sarcolemma becomes depolarized, which causes release of calcium ions from sarcoplasmic reticulum. With increase in the calcium concentration in sarcoplasm, calcium goes and binds with subunit of troponin, which causes unmasking of active site for myosin. Till that time, myosin head cannot bind because it is masked. Now unmasking occurs. Okay. After unmasking, next step, the ATP. Already we talked about this ATP site. Okay. After unmasking, ATP undergoes hydrolysis into ADP and phosphate, releasing energy. Using that energy, myosin head binds with exposed active site to form. After hydrolysis into this, using the energy, it binds with the active site to form cross bridge. So cross bridge formation occurs after hydrolysis of ATP, that too after exposure of the active site, unmasking of the active site. And now once cross bridge is formed, it swings towards the M line. See, it bends like this, pulling along with it thin filament forward. And when thin filament is pulled forward, other end it is attached to Z is also pulled forwards or towards the M line. And this is sliding or rotation along with the thin filament is pulled forward. And once it is done, ADP and PIR released. ADP, PIR released. Myosin head becomes relaxed. Now, new ATP comes and binds with. Because once these are released, this pocket becomes empty. So new ATP comes and binds. Binding of new ATP breaks down the cross bridge. So breaking of cross bridge. Again this ATP undergoes hydrolysis using that energy. Again it binds to another active site forming new cross bridge. The process, they are repeated in a cyclic manner like that as long as muscle receives signal. Is that clear? And during this process, thin filaments are sliding over. They are being pulled. And one more thing, in each thick filament, half of the myosins are directed towards one Z, other half to the other Z, so that they can pull the thin filaments from both the sides towards the center. Which band shorten? I band. H zone decreases. H decreases. Sarcomere? A band? Length of thin filaments? Length of thick filaments? Length of I band? Length of A band? Sarcomere length? Z pulled towards, almost sarcomere length becomes equal to length of A band. Fully contracted condition. Okay, next one. So the whole contraction needs hydrolysis or utilization of ATP molecules. That means energy supply is very important, and you know that ATP are released by oxidation, and for that oxygen is needed. If oxygen is not sufficient, then anaerobic breakdown occurs, forming lactic acid. Accumulation of lactic acid causes muscle fatigue, and the reaction time of muscle fibers vary. Different muscle fibers respond to the stimulus differently. And here we talk about relaxation. These crossbreed cycles continue to occur as long as signals are given to the muscle. Once nerve impulse is stopped, all calcium ions are pumped back to sarcoplasmic or cisternae of the sarcoplasmic reticulum, and in the absence of calcium, troponin again masks the, and once the active site is masked, cross bridges cannot be formed. So all the cross bridges, they move back to their. So Z move back, all move back to the original position. Muscle is said to be relaxed. So relaxation occurs when calcium ions are pumped back into cisternae. That happens when signal is stopped. Okay. And what is the oxygen binding special protein in the muscle? Myoglobin. It has greater affinity to bind with oxygen. Its oxygen binding affinity is much higher than that of hemoglobin. P50 for hemoglobin is 26.7. For this 2 point only. Very fast it binds with the oxygen. And depending on the amounts of these, muscles are classified into red muscles and red, white. Myoglobin, abundant, less. Mitochondria, less. Endoplasmic reticulum, less. More. What is the source of energy? Oxidation. So these muscles are oxidative muscles. Aerobic muscles. These are because glycolysis is the main source. Glycolysis does not need oxygen. So these, which muscles contract faster? White. Because sarcoplasmic is abundant, lot of calcium can be released immediately and muscle can contract very fast. Slow oxidative muscles are fast, glycolytic muscles are anaerobic muscles are aerobic muscles are which ones contract with low intensity? High intensity. Marathon race, long distance running, events like high jump, long jump, which need to occur high intensity. These are the done. Next, skeletal system. Skeleton gives support to the body, and skeleton is made up of only bones. Mostly bones and few cartilages. In the fetal stage, number of bones more than 300. Adult stage 206 and few cartilages. Cartilage has solid pliable matrix. Pliability is due to chondroid inserts. Bone matrix non-pliable, rigid due to deposition of calcium salts. And our skeleton is categorized into axial and appendicular. All those which are along the central axis, axial. Sternum. Vertebral column, ribs. They come under exoskeleton. Number of bones in exoskeleton, 80. And skull is the skeleton of the head region. It is formed by 22 bones. Formed by 22 bones, of which 8 are cranial, 14 are facial. Cranium is brain box, bones enclosing the brain. Total 8. Which ones are paid? Parietal, temporal, frontal, occipital, sphenoid and ethmoid. Keystone bone, because it articulates with all other bones. Occipital bone at the base has 2 occipital condyles, such as skull is dicondylic skull, articulate with the atlas or first vertebra. And then bones which form the face or support the facial part, facial bones, 14. All are paid except 2. Vomer and mandible. Largest and strongest, mandible. Largest facial bone, smallest facial bone, lacrimal. Smallest bone in the body, scales. Lacrimals, pair of nasals, 1 vomer. Inside the nasal chamber, inferior concave. Cheekbones are zygomatics, maxilla, upper jaw bone, mandible is lower jaw, palatines. These are the bones. And these bones form the skull. Okay. In addition to that, middle ear encloses 3 ear ossicles. Malleus, outermost is, innermost is, and the smallest one. So total number of ear ossicles, 3 pairs, both the sides. And hyoid. Hyoid is not actually a skull bone, but it has to be counted along with some structure in the exoskeleton. It is at the base of the buccal cavity, and this bone, U-shaped one, it doesn't articulate with any other bone in the body. Done? Next one, vertebral column. Location, mid-dorsal, and it is made up of serially arranged 26 vertebra. In the fetal stage, 33. Adult, 26. Because few vertebrae undergo fusion. And vertebra is a hollow structure enclosing a canal called, through which passes. That means vertebral column encloses and protects the spinal cord. It gives support to the skull region. It provides surface attachment of ribs, back muscles. Okay, so all. And main supporting part of the trunk region. Coming to the regions, first part is cervical. Number of vertebra, 7. Normally number of vertebra vary from species to species, but almost all mammals, cervical vertebra 7. Few exceptions are there, mostly 7. First one being atlas, which articulates with the condyles of the skull directly. Second one is axis. Axis forms a process called odontoid process, around which atlas rotates, causing rotatory movement. And then thoracic vertebra number, they are mainly specialized for articulation with ribs. Lumbar, strong, large vertebra number 5. Sacrum, 1. Fusion product of 5 sacral vertebra. Last one, coccyx, fusion product of. Next. Sternum. Sternum is called as breastbone. It is mid-ventral. It provides surface attachment of ribs as well as some abdominal muscles. It has manubrium, body, xiphoid process. Okay, mid-ventral, single bone. And ribs, 24 or 12 pairs. These are curved bones which dorsally articulate with which vertebra? Thoracic vertebra. Not any vertebra, thoracic, and ventrally with sternum through a cartilage called name costal cartilage. Type is hyaline, it's a costal cartilage. Through a cartilage, ribs are attached to the sternum. And dorsally they articulate with vertebra through 2 articulation process, capitulum and tuberculum. That's why ribs are called. Ribs are bicephalic, and they are classified into 3 groups. First 7 pairs directly attached to vertebra and sternum. So vertebro-sternal ribs, and they are the true ribs. 8, 9, 10 dorsally attached to vertebra, ventrally to cartilage of the 7th one. Chondro means cartilage. So vertebro-chondral. 8, 9, 10 attached to cartilage of the 7th one. Okay. And last 2, see, they are attached dorsally to vertebra, ventral side is free. 11 and 12, they are called or vertebral, because attached only to vertebra. Vertebral ribs are floating. Only first 7 are true ribs. That means remaining 2 categories are not true. So they are false ribs, both floating and vertebral and. Next one, limb skeleton. Appendicular skeleton, we completed axial. Total number 80. Appendicular skeleton 126. Each limb has, so all the 4 limbs 120. Pectoral girdle 4. Pelvic girdle 2. 126. Okay. And first one, pectoral girdle. Girdles give support to the limbs. They help in articulation of limbs with the exoskeleton. Pectoral girdle, 2 halves. Each half has 2 bones. Scapula, dorsal bone, ventral one, shape of scapula, flat triangular, broad flat triangular one, and it lies at the level of 2 to 7 ribs, 2 to 7, and dorsally it has an elevation or ridge called spine. Spine ends as a broad expanded process, acromion process, with which articulates clavicle. Clavicle is slender bone with 2 curvatures. One end articulates with acromion process of scapula, other end with sternum. And just scapula, just below the acromion process, cup-like cavity, glenoid cavity, with which articulates head of the humerus to form ball and socket joint. That's it. Next one, pelvic girdle. Pelvic girdle also has 2 equal halves. Each half has single bone called coxal bone. Coxal bone is formed by the fusion of ilia, ischium, pubis. At the point where the 3 fuse, cup-like cavity, acetabula, with which articulates head of femur to form ball and socket joint. Plus the 2 coxal bones of both the sides, mid-ventrally joined by fibrous cartilage called pubic symphysis. Cartilaginous joint. Okay. Next one. Limbs. 4 limbs. Tell me the names of the bones. Humerus, radius ulna, wrist bones are. Number. Palm bones are. Number. Challenges. Formula 2, 3, 3, 3, 3. 2, 3, 3, 3, 3. And correspondingly hindly bones. Humerus equivalent. Femur, longest bone, commonly called thigh bone. Then tibia or weak or feeble bone, fibula. Tibia is a strong one, actually weight bearing one. Ankle bones are tarsal. Number is 7. One less comparatively, but that is compensated by kneecap or patella. Covers the knee joint ventrally, cup-like bone. Yeah, ventrally this side, and it's a sesamoid bone formed by the ossification of tendons. And then metatarsals 5, phalanges 14. Same, no need to discuss. Okay, next one. Joint. Joint is the point of articulation between 2 bones only. No, bone or cartilage. Bone or cartilage. And here main point is joints act as and facilitate the movement. And if you consider the types of joints, okay, if 2 bones articulate by means of dense fibrous connective tissue, that means joint is formed by fibrous connective tissue joining 2 bones. They are called fibrous joints. Sutures, cranial sutures. Thin layer of connective tissue binds these flat bones. So, mobile or immobile? Immobile joints are called synarthrosis. Slightly mobile, antiarthrosis. Freely mobile, diarthrosis. Immobile or synarthrosis. Cranial sutures are the best example. And same thing, if the two bones are joined by cartilage, it is called cartilaginous joint. Two vertebrae joined by intervertebral disc made up of fibrous cartilage. So it's a cartilaginous joint. Okay, next one. Pubic symphysis between two halves of pelvic girdle is also cartilaginous joint. Then synovial. Synovial joint. In fibrous and cartilaginous joints, common feature is there is no cavity separating the bones. So they are either immobile or slightly immobile depending on the pressure, compression exerted. But unlike that, synovial joints are freely movable because the two articulating bones are separated by fluid-filled cavity called synovial cavity. It is bounded by synovial capsule and end of the bone lined by hyaline cartilage called articular cartilage. Articular cartilage. And synovial fluid acts as a lubricant, reduces the friction between the bones during the movements at the joint. Okay, these are the main points acting as a lubricant. And these are again categorized into ball and socket joint, shoulder joint between humerus and glenoid cavity, hip joint between acetabulum and head of femur, hinge joints, elbow joint between humerus, radius ulna, knee joint, interphalangeal joints between the phalanges also hinge, only angular movement in one direction. Third one, pivot joint. Pivot joint is between atlas and axis, radius and ulna, rotation of the arm and palm, upward and downward movement. So radius and ulna example. Next one, gliding. Gliding joint observed carpal, tarsal, zygapophysis of successive vertebra. Carpal, metacarpal of thumb, saddle joint. Skull and atlas, condyloid joint. Occipital condyles articulate with atlas, condyloid joint. When which joint is involved, saying no. Condyloid joint. Condyloid joint between skull and atlas. Between atlas and axis, pivot joint. Next one. Disorders. Autoimmune disorder. Myasthenia gravis, which affects neuromuscular junction. The receptors are blocked by the antibodies, which leads to fatigue, paralysis, weakness of skeletal muscle. Degeneration of skeletal muscle tissue, muscular dystrophy, most commonly caused by genetic. It's a genetic disorder. Rapid wild spasms, contraction, tetany, due to low calcium in body fluid, or blood or body fluids, not bone or muscle. In the body fluids, low. Hypocalcemia. Next, osteoporosis, bones becoming porous with the loss of calcium, and that occurs mainly due to decreased levels of estrogen, thus common in post-menopausal women. Plus, not only that, PTH, high secretion of PTH, more demineralization also can be caused by that. And then inflammation of joints is arthritis, inflammation of joints due to uric acid, gout disease, that is due to gout. And osteoporosis is subjected to even fractures also very easily. Next one. Discussed all the disorders? Did I miss anything? Gout I asked. You missed it, not I. Next. And anatomical unit is muscle fiber, functional unit is sarcoma. Next one, neural. Regarding neural system, first of all, coordination. Two or more organs interacting with one another and complementing one another's function is called as coordination. You are given that example. When you start running, actually muscle tissue, leg muscles are involved, but along with that to support that activity, heart rate increases, breathing rate increases, blood pressure increases, kidney function gets affected. That means many organs help to support that particular activity. This is called as coordination. It is brought about jointly by neural system and endocrine system. Neural system provides point-to-point contact, quick coordination. It occurs with impulse generation, which is very fast, occurring in milliseconds time. And regarding this neural, first neurons appeared in mid area, hydra, where the nerve cells are arranged in the form of a network, not very specialized. And with evolution, more and more complex it has become. Insects which fly in the air, they have well or ganglia, brain, nerves, fibers and all that. Definitely vertebrates are having much more well-developed neural system, divisible into CNS and PNS. CNS includes brain and spinal cord, protected the cranium and vertebral column, and all the nerves as soon as CNS come under PNS, cranial nerves and spinal nerves. This is having two types of fibers. Fibers which conduct towards CNS, efferent or sensory. Away from the CNS, efferent or motor fibers. And PNS is divisible into somatic and autonomic. Relay of impulses from CNS to skeletal muscles. To smooth muscles and involuntary. Autonomic. Autonomic subdivided into sympathetic and parasympathetic. In addition to these, another technology, basilar neural system, part of PNS only, but it is mainly concerned with conducting impulses to the visual organs from the visual organs. So all the nerves, fibers, plexus, ganglia involved in this conduction come under visual neural system. Done? Next, neural system made up of neural tissue, and you know that in the neural tissue two types of cells are there, neurons and which ones are the functional units? Neuron. It has three components. Easiest part. Cyton or cell body. It has a prominent nucleus. All cell organelles in the cytoplasm. Additionally, Nis's granules made up of ribosomes and rough endoplasmic reticulum, indicating that it is concerned with protein synthesis. And from the cyton, two types of fibers are given. Short branched fibers are dendrites. They carry the impulse always towards cyton. They also have Nis's granules. Single long process which conducts away from the cyton called as axon. Axon has no Nis's granule. Membrane of axon is axolemma. Cytoplasm is axoplasm. And point where axon starts is called axon hillock. And at the terminal end, it forms 5 branches called axon terminals or telodendrites. They end with swollen knobs, synaptic knobs containing synaptic filled with and axon can conduct impulse to another neuron or glands or other muscles, etc. Done? And neurons based on number of fibers, dendrites and axon classified. What is this? Bipolar. Only one axon is there, no dendrites. Total number of fibers, 1. So unipolar. Very rare, found in the embryonic stage. 1 dendrite, 1 axon. Totally 2 fibers, bipolar, retina and olfactory epithelium also. 2 or more dendrites and 1 axon, mighty. Most commonly multipolar cerebral cortex. Not only that, many neurons in our body are multipolar. And in addition to that, one more classification based on presence or absence of myelin sheath. In this case, axon is not surrounded by myelin. Myelin is a layer of lipids, fats, and produced by Schwann cells in PNS, oligodendrocytes in CNS. So myelin is formed by two types of cells, Schwann cells and oligodendrocytes. And in this case, this is the Schwann cell. You can see the nucleus of the cell also. Schwann cell produces that myelin. Is it continuous? No, it is discontinuous. Gaps between two myelin sheets called as nodes of Ranvier, and myelinated appear white in color. They are found in cranial nerves and spinal nerves. If myelin sheet is not there, unmyelinated, because there are no Schwann cells. Schwann cells are present but do not secrete the myelin, and unmyelinated are found in somatic and autonomic neural systems. Done? Next. Generation of nerve impulse. Actually, nerve cells are excitable cells. Why they are excitable? Because they are polarized condition. Why they are polarized? Because of difference in the permeability and difference in the distribution of ions. We are first discussing resting membrane. Resting membrane is permeable to, nearly impermeable to. So permeability is not same for all. That is one reason. Second reason, sodium ions outside 10 times more than that of inside. Potassium 30 times more than outside. Plus axoplasm has negatively charged proteins which cannot move out because membrane is not permeable to them. Okay. So at the end, if you consider, ECF has high concentration of sodium, low concentration of axoplasm has low sodium, high potassium, high proteins also. Okay, so due to this differential distribution, finally it ends with positive charge outside and negative charge inside. This condition is polarized condition, and the difference in the charge across the membrane, potential difference, is called as resting potential or resting membrane potential, which is around minus 70 millivolts. Okay, and then this is a normal condition. That means concentration gradients are there for sodium and potassium in the resting membrane, which is one of the reasons for polarized condition of the membrane. These gradients must be maintained. Done by sodium potassium pump. Each sodium potassium pump, also called sodium potassium ATPase, both the words are. Each pump pumps 3 sodium outwards in exchange for 2 potassium inwards. By pumping, passively or actively? Actively, it helps to maintain those gradients. So far we discussed resting condition. Now coming to the stimulus generation, whenever a stimulus is given, changes occur only at the point of stimulation, not the total axon. Now let us name this as A. When a stimulus is given, normally membrane is nearly impermeable to sodium. But when a stimulus is given, it induces the membrane to become more permeable to sodium. Now sodium influx occurs. Sodium rapidly moves inward. Which charge is moving in? Positive. That means now inside becomes positive, outside becomes. That means there is reversal of the charges, and now the membrane is said to be depolarized. And it happened due to what? Influx of sodium. And in a depolarized membrane, the potential difference across the membrane is called action potential, which is nothing but nerve impulse. Here it is resting potential, now the depolarized state action potential or nerve impulse. Okay, and when A part is depolarized, adjacent region B is in or resting condition, due to which there is difference in the charges. Here inside has become, but adjacent region, when there is difference, current flows from positive to negative. So there is flow of current from A to B along inside, B to A along outside. This flow of current changes the membrane permeability of B region. Now influx of sodium occurs and now it starts becoming. Same manner, depolarization of B causes depolarization of C. Like that, it is self-propagated till the end of axon. Is that clear? And once impulse is generated, for some time, it cannot generate the next impulse because it has to come back to its original state before it is able to generate. That period is called as refractory. That's why impulse cannot travel backward, always travels forward only towards the axon. Now coming back to the next point, when A becomes depolarized, automatically it causes depolarization of B. A has become depolarized due to sodium, and this influx of sodium is induced by stimulus. Stimulus induced increased permeability to sodium is extremely. Once again it becomes more permeable to potassium and it causes outward movement of K plus with the efflux of K plus. No movement of sodium, don't mention about it, okay? When we talk about it specifically, take that point only. So when K plus goes out, once again outside becomes positive, inside becomes negative. That means it's coming back to its original condition, which is called as repolarization. So repolarization is due to efflux of potassium. Depolarization is due to influx of. Any doubt? That's it. Next one. Transmission of impulse. Actually, synapse is a junction between two neurons, and with respect to that synapse, the membranes are called as presynaptic and postsynaptic. Presynaptic neuron, presynaptic membrane, postsynaptic membrane are in close proximity here, not separated by synaptic cleft. Unlike that here, this is presynaptic, this is postsynaptic, there is a fluid filled cavity separating them. So, synapses are of two types. Conclusion. A synapse is formed by presynaptic membrane and postsynaptic membrane with or without synaptic cleft. If there is no cleft, that means membranes are close proximity, electrical synapse. If there is a gap, chemical synapse. So there are two types of synapses. Electrical synapse. Impulse is conducted as we discussed earlier, B to C, C to D, like along the length. Once impulse reaches the presynaptic membrane, as there is no synaptic cleft separating it from postsynaptic, electric current flows directly from presynaptic to postsynaptic. That means impulse conduction across this synapse occurs in the form of electric potential only. So they are called electrical synapses. And it occurs same as the process occurring along the length of axon and very fast, quick, no time delay. It's a continuous activity. And these synapses are rare. It may allow bidirectional conduction also. Done? Second one, chemical synapse. In this case, presynaptic, postsynaptic separated with fluid filled synaptic cleft, and through the cleft or gap, current cannot, current needs conductor, isn't it? That's why electrical is not possible. So they use a chemical substance for the purpose, neurotransmitters. So they are called chemical synapse. Now let us take the sequence of events. Nerve impulse reaches the axon terminal. So the terminal membrane becomes depolarized. When the membrane becomes depolarized, this step is not there in the NCERT. We discussed last week, you were given a question on that. When the membrane becomes depolarized, calcium channels of the membrane open, calcium enters into the synaptic knob. With increase in the calcium concentration, the vesicles move towards the membrane, fuse with the membrane to release neurotransmitter by exocytosis. Is that clear? Calcium concentration increases. This only causes the vesicles to move towards the membrane, fuse with them and rupture to release the neurotransmitter. That's why when they ask a question, some chemicals block the calcium. Without calcium, can vesicles move and release? So, neither neurotransmitter is released, nor without neurotransmitter, postsynaptic neuron cannot undergo any change. Both are not possible. Okay, now coming back. What is the most common neurotransmitter? What is this membrane? Postsynaptic membrane. It has receptors for neurotransmitter. So, neurotransmitter binds to the receptor. Binding causes opening of some ion channels. With ion channels opening, ion movement occurs and a new potential is developed in the postsynaptic membrane. We call them as postsynaptic potentials. They may be excitatory or inhibitory. If the neurotransmitter causes depolarization of the postsynaptic membrane, it may be excitatory. If it causes hyperpolarization, making it more negative, inhibitory. Done? Next. CNS, central neural system, only brain is discussed here. Anyway, first paragraph, all the functions brain listed. Central, commanding, controlling, processing unit, you can use any of these. Almost all functions which we discuss later are summarized there. So just check that paragraph later. Now, brain is enclosed in the cranium, surrounded by three meninges. Which one is unique to mammals? Arachnoid. Outer double-layered fibrous dura mater. It forms inner lining of cranial cavity. Outermost covering of brain forms lining for the cranial cavity. Middle arachnoid, thin spider web-like. Inner pyometer. Which one is adhered to surface of the brain? Pyometer. Lining of cranial cavity? Dura mater. Unique to mammals? Arachnoid, middle one. Okay. And the three regions are forebrain, midbrain and hindbrain. And in this diagram, almost all this is cerebrum only. See, the largest part, cerebrum. Second largest is cerebellum. Okay. Now, coming to the description, forebrain includes olfactory lobes are there. Remember that point. They are the extensions of limbic system, brain's limbic system. Cerebrum, largest part. It is divided by longitudinal fissure into two cerebral hemispheres. Each hemisphere has frontal, parietal, temporal, occipital lobes, and the two hemispheres are joined by transverse tract of fibers called as corpus callosum. If they ask myelinated or unmyelinated, myelinated. The fibers are myelinated only. And so information dissipates between right and left ones. And the surface of the cerebrum is highly convoluted, forming gyri and sulci, increases the surface area to accommodate more number of neurons. And it is covered by layer of cells called cerebral cortex or neopallium. This is also called grey matter because it has concentration of cell bodies of the neurons. Cell bodies are concentrated here. And there are three functional regions. Cerebral cortex, grey matter, it is sensory area, motor area, association area. Sensory area, sensory function, motor area, motor. Association, neither clearly sensory nor motor and is concerned with intersensory memory and communication. Memory and communication. And then inner part is cerebral medulla. It is white matter because it contains myelinated fibers. So it is white in color, called as white matter. and next part. After that, cerebrum wraps around. Thalamus, one point is there. Major coordinating center for sensory and motor signaling. Sensory and motor signaling. And next, in the thalamus, the basal part is called as hypothalamus. Hypothalamus has many centers with neurosecretory cells that secrete hypothalamic hormones. It has centers for thermoregulation, body temperature, thermostat of the body, and urge for drinking and eating. Those centers are also there and also produce many hypothalamic hormones. Now coming to the end part, inner parts of cerebral hemispheres, some deeply located structures like amygdala and hippocampus together form limbic system or limbic lobe. Limbic system along with hypothalamus, olfaction, autonomic responses, regulation of sexual behavior, expression of emotions. Next, midbrain. Midbrain lies between thalamus or hypothalamus of and pons of the hindbrain. And midbrain, 2 points. Running to the center of midbrain, there is a canal called cerebral artery. Okay. Dorsal side of the midbrain, 4 optic lobes are present. Corpora quadrigemina. Crura, cerebral, ventral side. Okay, additional points. And regarding this summary point, it integrates auditory, visual, tactile. Auditory, visual and tactile means touch. Next one, hindbrain. 3 parts. Pons, cerebellum. Pons only 1 point. It has a number of nerve fibers that interconnect different regions of the, because it is such a vulnerable position, any information coming from forebrain, cerebellum or spinal cord, medulla, everything has to pass through this. So it interconnects many regions. And cerebellum, second largest part, called as little brain, and this has outer grey, inner white matter. That means you can call cerebellar hemispheres with cortex and medulla. White matter, inner white matter is projected into tree-like branches called arborvitae. Arborvitae. Cerebellum surface also highly convoluted to accommodate more number of neurons. Cerebellum is a center which maintains balance or equilibrium, so it acts as gyroscope of the body, and it also receives inputs from. Ear also is meant for balance. So semicircular canals and auditory system. Last one, medulla. Medulla has centers for all involuntary respirations, cardiovascular center, gastric secretions, vomiting center, all involuntary centers. And of this, if you look at the diagram, as if the whole structure, main part of the brain resting on this stalk-like structure, and this stalk is formed by midbrain, pons, medulla. Cerebellum is not along the central axis; it is towards the dorsal side. Okay. So, which part of hindbrain not involved in brainstem formation? So, midbrain and pons, medulla together constitute that. Next one, chemical coordination, and regarding that, chemical coordination, neural coordination jointly regulate all activities or coordinate the body functions. Here you need to learn the definitions. Who is the father of endocrinology? What is the first hormone? Secretin. And hormones, what is the need to have again endocrine system, another coordinating system? Nerve fibers do not innervate each and every cell, but every cell activity must be regulated. That's why we have another system, but work in the same manner, jointly bring about the coordination only. And these are ductless glands which secrete hormones. Hormone classical definition: They are produced by classical old endocrine glands, released into the blood, transported by blood to target, distant target. But now we have a modern definition. Non-nutrient chemicals which act as intercellular messengers and trace hormones. This definition covers not only the hormones produced by endocrine glands but by other non-endocrine structures also. So this is more appropriate. Next one. Endocrine system is less developed in invertebrates, includes only few hormones or few structures, but well developed in all vertebrates. And endocrine system includes not only well organized endocrine glands like pineal, pituitary, thyroid, parathyroid, thymus, adrenal, pancreatic islets, gonads, other than the non-endocrine structures, gastrointestinal tract, heart, kidneys, they all these come under this description. What is the first part we discussed there? Hypothalamus. Hypothalamus is actually basal part of the thalamus. It's present in the brain and it has groups of cells called neurosecretory cells. Hormones produced by the cells released at the axon terminus. Some hormones, they are released directly into blood circulation. Capillary network present in the hypothalamus. They join to form blood vessel, which again ends with capillaries in the adenohypophysis. So the hormones are transported via portal circulation to the adenohypophysis or anterior pituitary. And some axons are, that is neurosecretory cells are having long axons which extend directly into neurohypophysis. Okay. Question. Hypothalamic hormones reach neurohypophysis via axon, portala. That is the main point. Hypothalamic hormones reach the neurohypophysis axonally, directly by axons. Axons are nerve fibers. Actually, bundle of nerve fibers is called tract. Hypothalamo-hypophyseal tract, directly by axons, not by blood. Hypothalamic hormones reach the adenohypophysis via blood circulation, that is portal circulation. And these hormones which reach adenohypophysis via portal circulation are of two types. Releasing hormones are through those hormones, it regulates the synthetic activity of the adenohypophysis. Whenever releasing hormones reach, releasing hormones stimulate the antipituitary synthesize and secrete the hormones. Inhibiting hormones stop the secretion. That means all pituitary, anterior pituitary hormone synthesis directly regulated by neurohypophysis does not synthesize anything. It just receives the hypothalamic hormones and releases them into blood circulation directly. That's why it is under direct neural regulation of hypothalamus. That's it. Okay. Example, GnRH, gonadotropin releasing hormone, stimulates anterior pituitary to release gonadotropins. Thyrotropin releasing hormone, stimulates thyroid, stimulates TSH secretion. Thyrotropin secretion, not TSH secretion from that. So like that, and somatostat. Growth hormone, inhibiting hormone. Growth hormone, releasing hormone, somatotropin. Growth hormone is somatotropin. Somatotropin. Now coming to the next part, that's all about it. Now coming to the pituitary. Pituitary is lodged in Tarsica. Depression in the sphenoid. Pituitary gland has anatomically two divisions, adeno and neuro. Adenohypophysis has two, pars distalis, pars intermedia. Pars nervosa or posterior lobe is neurohypophysis, and they secrete various hormones. First one, pars distalis or anterior lobe, it secretes growth hormone or somatotropin. It stimulates growth of various tissues including bones, muscles, everything. And growth hormone deficiency in the childhood causes stunted growth, dwarfism, pituitary dwarfism. Excess secretion Gigantism. Person grows very tall. And excess secretion in the adult stage, acromegaly, disproportionate growth, as person cannot grow in height because growth centers of bones are closed, epiphyseal plates are closed, and excess growth hormones stimulate other tissues to grow. So thickening of soft tissues, abnormal thickening of the bones, length cannot increase, thickening occurs. And this acromegaly not easily detected in the beginning stages unless disfigurement occurs in the facial region, and if it is not, so it is left like that undetected for long time, if it is not diagnosed, it can cause premature death also, may be dangerous. That is acromegaly, excess secretion adult stage. Next one. Okay, TSH, thyroid stimulating hormone or thyroid tropin, which stimulates thyroid gland to synthesize thyroid hormones. ACTH, adrenocorticotropic hormone or corticotropic. It stimulates adrenal cortex to secrete corticoids. PRL, prolactin, stimulates growth of mammary glands and milk secretion. Next. Gonadotropin. Gonadotropins are of two types, FSH and they stimulate gonads, so together called as gonadotropin. Follicle stimulating hormone. In males, it stimulates Sertoli cells to secrete some factors which help in spermiogenesis. And FSH in females stimulate growth of the ovarian follicles. And LH, luteinizing hormone. In males, it is also called ICSH, interstitial cell stimulating hormone. Stimulates Leydig cells to secrete androgens which help in spermatogenesis. LH in females cause ovulation, rupture of the Graafian follicle, formation and maintenance of corpus. Okay. Next one, pars intermedia. This is actually merged with pars distalis and it secretes only one hormone, MSH, other name melanotropin. It stimulates pigmentation. Melanocytes are stimulated, causing pigmentation. And then posterior pituitary releases just two hormones synthesized in the hypothalamus. ADH already we discussed. Reabsorption of what? The distal tubule, preventing diuresis, and also can cause that's why it's called vasopressin, other name. And deficiency causes diabetes, excessive water loss in the urine. And then oxytocin, it has effect on smooth muscles, contraction of smooth muscles, especially uterine muscles in childbirth or parturition. Plus ejection of milk, not only uterine contractions, ejection of milk. Next one, pineal. Pineal gland is present attached to dorsal side of the forebrain, and this gland secretes a hormone called melatonin derived from tryptophan amino acid, and this regulates 24-hour rhythms, diurnal rhythms, sleep-wake cycle, body temperature, pigmentation, metabolism, menstrual cycle, defense capability. Next one, thyroid. Largest endocrine gland is thyroid. It has two lobes on either side of the trachea. The two lobes are joined by connective tissue called as isthmus. Each lobe is made up of many follicles. Each follicle is lined by follicular cells that secrete two hormones, T3 and T4. Most abundant thyroxine is, most potent is T3. T3, potent, not abundant, not quantity. Potency means effectiveness. So they are interconvertible, T3 and T4. And 3, 4 iodine, that's the difference. Triiodine, tetraiodine. That means iodine is essential for production of these hormones. So iodine deficiency can cause enlargement of thyroid gland, a condition called as simple goiter. And deficiency of thyroid hormones during pregnancy and the newborn can result in a disorder called cretinism, where physical and mental growth are retarded, IQ levels very less, abnormal skin, deafness and mutism are common symptoms. And deficiency in the adult stage causes mixed edema. Edema is the main symptom, that is puffiness, fluid accumulates in the tissues, and metabolic rate decreases, lethargy occurs, intolerance to cold. The only point mentioned in the book, menstrual cycles become irregular in females. Okay, and excess secretion causes Graves' disease, hyperthyroidism, exophthalmic goiter, because typical symptoms, protrusion of eyeball, enlargement of the thyroid gland. And in Graves' disease, BMR is very high. So weakness, weight loss, heat production also is very high. What? Next one. On the back side of each lobe of thyroid, 2 glands are present, parathyroid. Total number 4. Parathyroid gland produce PTH, parathyroid. Thyroid produces TCT, nature, protein. TCT is hypocalcemic hormone, decreases the blood calcium. PTH is hypercalcemic hormone. How does it increase blood calcium? Promotes demineralization or dissolution or resorption, bone resorption. Second one, promotes reabsorption of calcium in the renal tubules. Third one, absorption from the food, right, with the help of calcitriol, active form of vitamin D. And what kind of hormone is PTH? Chemically, peptide. TCT is protein, it is given as peptide hormone, and these two are antagonistic. One more important, it is not controlled by pituitary. Circulating levels of calcium regulate the production of these. Next one, thymus. Thymus is a lobular gland present behind the sternum, ventral to the. Able to recollect all? Yes. And thymus produces thymosins, peptide hormones. These hormones cause differentiation of T lymphocytes, thus helping. They stimulate B lymphocytes to produce antibodies and helping humoral immunity. Thymus size gradually decreases and in old age degenerates. That's why immunity becomes very weak in the old age. Next, adrenal. Adrenal gland above the kidney, not anterior. Present above the kidney, superior or above the kidney. Adrenal gland has 2 different parts, outer cortex and inner medulla. Medulla is ectodermal, cortex is mesodermal in origin. Adrenal medulla secretes 2 types of hormones, epinephrine and derived from thyroxine, amine, that's catecholamines, and these are produced in response to stress or emergency situation. So they are called emergency hormones or epinephrine and norepinephrine. Increase alertness, pupillary dilation, pyroruption, sweating, heart rate, breathing rate increases, heart rate increases, strength of contraction of heart also increases. Next, breakdown of glycogen. So blood glucose level increases. Proteolysis and glycogen. Glycogen, protein, lipids all break down. So nutrients availability is increased in the blood. That's it. Next one. Adrenal cortex. Cortex has 3 regions. Outer glomerulosa, middle glomerulosa, fasciculata, reticularis. Glomerulus, all hormones of cortex are steroid hormones called as corticoids. They are grouped into 3 based on their function. Regulating glucose metabolism, mineral metabolism, sex characters, secondary sexual characters. Zona glomerulosa produces Principal one is aldosterone. Aldosterone promotes reabsorption of sodium, water, secretion of platelets and phosphate ions. Okay, and thus maintains the mineral balance. And by causing absorption of sodium and water, it affects blood volume also, blood pressure also, osmotic pressure also, isn't it? More water absorbed means blood volume, blood pressure also increases, osmotic pressure decreases. Like that. That means not all through these electrolyte balance, these activities also controlled by mineralocorticoids. And fasciculator secretes mainly. Principal one is cortisol. They increase blood glucose level, promote gluconeogenesis, proteolysis, lipolysis. Next, inhibit cellular uptake and utilization of amino acids. Has effect on cardiovascular system and kidney functions. Support, especially cortisol, erythropoiesis. We didn't mention thyroid functions, by the way. Okay, I'll come back to that. And not only that, anti-inflammatory, suppress the immune reaction. That's why in allergy and all those to reduce, this can be given as a treatment. And the next one, sex corticoids. Sex corticoids are responsible for secondary sexual character, facial hair, axial hair, pubic hair at the age of puberty. So that is about adrenal. Adrenal corticoids, less secretion causes Addison disease, weakness, hypoglycemia, blood sugar level decreases. I think in the beginning itself it is mentioned. Under secretion means less secretion only. Carbohydrate metabolism, acute weakness, fatigue leading to disease called Addison's. Excess secretion, Cushing's syndrome. Okay, that is about it. Now let us discuss the thyroid. Thyroid hormones are mainly meant for BMR. Plus supports erythropoiesis, electrolyte water balance in the body. That's why cretinism, mental retardation occurs. Next one. Pancreas. Pancreas is a mixed gland or compound gland with both exocrine and endocrine portions. And what percentage is endocrine in nature? 1 to 2%. And endocrine portion includes 1 to 2 million clusters of cells called islets or Langerhans. They have 4 different types of cells: alpha, beta, delta, F cells. We are given only alpha and beta. Alpha cells secrete, beta cells secrete, and both are antagonistic to each other. Together maintain glucose homeostasis or balance. Now, glucagon, what cell secrete? Alpha cell. Alpha cell secrete. Nature, peptide hormone. Target cells, hepatocytes. And what are the effects? It promotes glycogenolysis, breakdown of glycogen to glucose. Glucose level increases. Gluconeogenesis, formation of glucose increases. Reduces cellular uptake and utilization of glucose. So it remains in the blood only, because cells are not taking it up. Thus it promotes. Okay. It can be called hyper. Which conditions stimulate the release of it? Hypoglycemia. Beta cell secrete. That is also peptide. Target cells are hepatocytes and adipocytes. And it is hypoglycemic hormone. It promotes cellular uptake and utilization of glucose. Glycogenesis converts glucose to. Prolonged hyperglycemia leads to. Due to prolonged hyperglycemia, sugar starts coming out along with urine, a condition called as glycosuria. And in the absence of insulin, cellular uptake of glucose does not occur. So cells deprived of glucose start using fats. Fat metabolism forms ketone bodies, ketonuria is a result. And diabetes mellitus is due to deficiency of insulin or resistance to insulin also. Summary it is given. That means in the person body, insulin may be produced normally, but cells are not able to respond to that. Next one. Testis. Testis is the primary male gonad. It acts as dual organ. It forms gametes like a primary sex organ, secretes hormones like endocrine. Both the functions, remaining points you know, lies outside the abdominal cavity in the scrotum, and it has seminiferous tubules and interstitial. In the interstitium are present Leydig cells. Leydig cells secrete androgens. Principal androgen is testosterone. What are the functions? Development, maturation and functioning of accessory or secondary sex organs. Development of, that means all those, prostate, vas deferens, seminal vesicles. Development of secondary sexual characters. That is at the age of puberty, changes. Change in the voice, low pitch voice, aggressiveness, broadening of shoulders, facial hair growth, all these things. Then stimulates spermatogenesis, more muscular growth of spermatogenesis, and then shows effect on CNS, responsible for male sexual behavior or libido, and stimulates anabolism of proteins and erythropoiesis. Stimulates the erythropoiesis. Then ovary. Ovary is also dual. Primary sex organ producing ova, also endocrine secreting hormone. And they are abdominal. Inside the ovary, two types of steroid hormones are produced, estrogen and estrogen secreted by growing follicles and affects development and functioning of accessory ducts and glands. Then secondary sexual characters, high pitch voice, breast development, etc. at the age of puberty, and growing estrogen stimulate further growth of the follicle into mature follicle, that is help in oogenesis process. Next, female sexual behavior, right? Next, after ovulation, remnants of ruptured follicle develop into corpus luteum. Corpus luteum secretes mainly progesterone. Progesterone is a hormone of pregnancy, prepares the uterine endometrium for implantation and maintains the pregnancy. Other than that, it also stimulates formation of alveoli in the mammary glands and stimulate milk secretion. Next, hormones are classified based on chemical nature. Peptide, protein, polypeptides. Pituitary hormones, hypothalamic hormones, all those. Other glands? Heart, no need to discuss. Just now we discussed A and N. Kidney, erythropoietin, RBC production. Gastrointestinal. Gastrin stimulates gastric glands to secrete pepsinogen and secretin. Stimulate exocrine pancreas to secrete water and bicarbonate ion. CCK stimulate pancreas to secrete pancreatic enzymes and gallbladder to release bile. So CCK affects both pancreas and gallbladder. Pancreas is affected by two hormones, secretin and CCK, and GIP, gastric inhibitory peptide, also called enterogastron, old name, that inhibits gastric secretion and gastric motility. Other than that, many tissues in our body produce growth factors which help in repair, regeneration and growth of the tissues. Now coming to this structural, chemical structure, we talked about peptide now. Amine hormones example, epinephrine and norepinephrine derived from tyrosine, and melatonin also derived from tryptophan. Iodothioneins T3 and T4, steroids, sex hormones and corticoids are steroids. Okay. And in addition to that, solubility, water soluble and lipid soluble. Steroids and iodothioneins, they are fat soluble. They enter the lipid bilayer of the cell membrane and bind to intracellular receptor. Others cannot enter because they are water soluble. They bind to Membrane bound receptors. See now look at this one. This is example. How many types of receptors are there? Two. Cell membrane, membrane bound receptors. Inside the cell nucleus also intracellular receptor. And FSH. It's a protein hormone. That means water soluble or it cannot pass to the lipid. So it first binds to receptor, and you remember the points. Receptors are very, very specific. They are present only on target cell. That's why hormone is able to show effect only on target cell. All receptors are nothing but proteins. All hormones are not proteins. They can be of anything. But all receptors are proteins. So first hormone binds with the receptor to form a hormone receptor complex. And actually this hormone needs to cause some change inside the cell, but is not able to enter the cell. So it stimulates formation or generation of second messenger in the cell, cyclic AMP, calcium, IP3. Normal, not FSH, in general 3 types of second messengers. And the second messenger now stimulate biochemical process, which in turn result in physiological. FSH stimulate growth of the ovarian follicles. Ultimate physiological effect is ovarian growth. And for growth, some proteins must be produced. That is biochemical process. And the next one, lipid soluble. They can enter the lipid bilayer of the cell membrane and they bind to the intracellular receptor, iodotyronins and steroid hormone. See, hormone directly enters the cell and binds receptor present inside the nucleus. Inside the nucleus, genome is present. Once hormone binds receptor, forming hormone receptor complex, it interacts with the genome or stimulate the chromosome and activate a particular gene to express. Gene expression first step, mRNA, transcription, mRNA is formed. It undergoes translation to form protein. Using that physiological response. Clear? So directly gene expression or interaction with the genome is observed in the case of intracellular receptor. Examples are sex hormones are steroid. Iodotyronins and steroids. Steroids include sex hormones and corticoids. Next one. That's it. Structural organization. I have 15 minutes time. Structural organization. I have class. Huh? Structural organization. I think tissues, cockroach is there. In this book it is there. I am not asking in the syllabus. Okay, what's a tissue? Group of cells. Only cells. Group of cells along with the intercellular substance constitutes a tissue. Study of tissues is histology. And tissues are of different types in different organisms, but basic types are 4. Animal tissues: epithelial, connective, muscular, neural. Epithelial tissue usually forms covering of the organs or body, forms lining of the tubes, ducts or hollow structures inside the body, and epithelial tissues are avascular, do not have direct blood supply. Cockroach is there? Only fraud. Okay, open the frog, I will explain frog. First let us take tissues then. Go back. If earthworm is there, tissues also would be there. Okay. So first introduction, animal tissues, the types of tissues are there. Now coming to epithelial tissue. Epithelial tissues are arranged compactly with intercellular substance literally negligible. Cells are arranged very compactly with little intercellular substance. Cells rest on basement membrane, no direct blood supply. They depend on underlying connective tissue for their nourishment, and they are classified in two types, simple and compound. Cells arranged in one layer, two or more layers. Compound is also called stratified. Okay. Simple epithelia, a single layer, helps in secretion, absorption, all that. Compound helps mainly in protection, and simple epithelia is further classified based on shape of the cells. Cells are thin, flat, tile-like squamous. As they are very thin, it forms a diffusion boundary. Exchange of substance occur very easily across that epithelium, so it forms a diffusion boundary. It has cells have irregular boundaries. It is found in alveoli, Bowman's capsule, line endothelium of blood vessels. All these are examples for simple squamous. Cells are cube shaped, cuboidal. Cuboidal found in the lining of ducts, tubular portions of nephrons, germinal epithelium of ovary, etc. And these are for PCT, DCT tubular portions. Free ends may bear microvilli forming brush border. Example, PCT. And then cells tall, slender, with a nucleus towards base. That is called columnar epithelium. And columnar epithelium is found lining of stomach and intestine. The free ends may bear microvilli, increase the surface area of absorption as in intestine. And fourth type, ciliated. Ciliated epithelium cells may be columnar or cuboidal, but free ends bear hair-like structures called as cilia. Cuboidal or columnar. Cuboidal, columnar both help in secretion and this is diffusion boundary. These two help in secretion or absorption. Ciliated epithelium may be cuboidal or columnar, free and bear cilia. Cilia cause movement in a specific direction. Example, bronchioles, fallopian. One more type, pseudo-stratified. Cells arranged in single layer but due difference in the height and position of nucleus appears as stratified but not. So pseudo-stratified trachea is the example. Okay, then the next one, glandular. Glandular cells are glandular epithelial cells. Again, may be cuboidal or columnar. They are secretory nature, so we call it as glandular. There are two types, unicellular and made up of single cell. Example, goblet cell, which secrete mucus in the gastrointestinal tract. A group of cells, aggregation of cells, multicellular. Most of the glands in body are multicellular only. And they are again classified into exocrine and glands with ducts are exocrine. All mammary glands, sweat glands, sebaceous glands, salivary glands, pancreas, all these are exocrine. Ducts carry out the secretion. Without ducts, and they secrete hormones which are released, diffused into the surrounding fluid, tissue fluids, because there are no ducts to carry out. Plus, remember, merocrine, apocrine. If the secretion is released without any loss of cellular material, merocrine, pancreas. Along with the secretion, apical portion is lost, apocrine. Complete loss, sebaceous glands. Example. Okay, next one. Compound epithelia. Compound is two or more layers and it is found in dry surfaces like skin, moist surfaces like buccal cavity, pharynx, vagina, etc. It has limited role in secretion and absorption, mainly protection from mechanical or chemical stresses. And it also forms lining of salivary ducts and pancreatic ducts. Next, how many types of cell junctions are there? Yes. All tissues, especially epithelial tissues, almost have all types. Those which prevent leakage of substances, which form cementing between neighboring cells, which form cytoplasmic connections, which ones allow transport of ions and molecules, which ones are compared plasmodesmata. Also called communication junctions, because adjacent cells communicate with each other through cytoplasmic connections. Next one. Most abundant tissues are connective tissues, and these tissues differ from epithelium in having Matrix well developed. Intercellular substance is very well developed. Cells are separated and in why are they called connective? Because they connect different tissues or organs in the body, most abundant. These are classified into different types from soft tissues to specialized connective tissues. Different types are there. All connective tissues except the blood, they have fibers made up of collagen and elastin. They provide support, strength, flexibility and elasticity to the tissue except blood. All connective tissues are vascular except cartilage. These two are the exceptions. And those fibers are produced by cells called fibroblasts, and these cells also secrete some modified polysaccharides which form the ground substance or matrix. And the types: loose connective tissue, it includes areolar and areolar tissue is again most widely distributed. It has mast cells, fibroblasts, macrophages. Macrophages are phagocytic. Mast cells produce histamine and inflammatory mediators. Fibroblasts produce the fibers. And areolar tissue occurs beneath the skin. Adipose is specialized tissue as well as loose connective tissue. Because loose connective tissue matrix is semi-fluid with cells and fibers loosely arranged. Same structure is there, but this tissue is specialized to store fats. Excess of nutrients get converted into fats and stored. So it is specialized tissue also. And next one, cells and fibers densely packed. It is called as dense connective tissue. If all the fibers are oriented along the same direction, regular, they are of two types, tendons and tendon connect bone to which muscle? Ligament connects bone to bone. Both are examples for dense, regular. If the fibers are oriented in different directions, irregular, found in the skin. Next one, skeletal tissues. Cartilage and bone are called as skeletal tissues because they form the skeleton of the animals. Cartilage is having pliable, solid matrix which can resist the compression. Already we talked, chondroitin salts are present, and the matrix is produced by cells called chondrocytes which are enclosed inside the lacuna. Space is called as lacuna, and collagen fibers are present inside the matrix. Found in tip of the nose, outer ear joints, limb bones, costal cartilages, all these are examples. And then bone tissue. Bones are highly vascular compared with the cartilage is avascular, bones are highly vascular, and in this also the non-pliable condition is due to calcium deposits, very hard and rigid. Cells are called osteocytes. They are also enclosed in lacuna, and they also matrix contains collagen. That is common. Okay, no elastic fibers, only collagen can be seen in this. And typical one, Haversian systems are characteristic. This is one Haversian system. This is Haversian canal, Haversian system, another Haversian system. All these are the Haversian systems characteristic of bone. And actually, they mentioned the bones. Bones give support to the body, protect the inner delicate organs, weight-bearing structures, and some bones, bone marrow produces blood. All those functions are listed. Blood we already discussed. Next, muscle tissue. Muscle tissue, already I mentioned these points. Contractility, elasticity, plus shortening, lengthening in response to stimulus. So, locomotory movements, all these are caused by the muscle tissue. These are of three types: skeletal muscles. Just check the points. Example, biceps, arranged in the form of bundles, held together by fascia. Already you have learned that. And elongated and cylindrical. They show alternate dark and light band appearance. That's why they are called as striated. Smooth muscle fibers, again, they are held by cell junctions and are bundled to form the muscles. And they are fusiform or spindle shape, multinucleated, uninucleated. Peripheral nuclei, central nucleus, and no striations because actin myosin are irregularly distributed. And then cardia, short, cylindrical, branch, single central nucleus. They also show striations and they have Intercalate. Junction between adjacent cells, plasma membranes are fused. They form intercalated discs, due to the presence of which they can contract as one unit. Meaning, when a signal is given to one muscle fiber, not only that fiber, all the neighboring fibers also contract at a time as one unit. That's why it can be described as functional syncytium. But structurally syncytium is next one. Neural tissue, nothing is there. Two types of cells, neurons and neuroglia. More than 50% is neuroglia. They do not have neural property, cannot generate impulse. They are just supporting cells. They give support and protection to the neurons. And regarding conduction, they just mentioned one point. Whenever a signal is stimulus is given to the neuron, a disturbance is generated in it, which travels along plasma membrane, and at the output zone, output means axon terminals, it is passed on to the next neuron or other tissue. You know that tissues of same function grouped to form organs, organs are grouped to form organ systems, and we are going to discuss the organ systems of cockroach and frog. Cockroach scientific name Periplaneta. Americana, general structures 1/4 inch to 3 inches, all the sizes given, red colored, yellow colored, green colored insects and all that. So, periplaneta 34 to 53 millimeters and all. Body divisions are, let us focus on main point. I don't think they ask 53 millimeters, 54 in a NEET exam. Head, thorax and abdomen. And cockroach body is covered by chitinous exoskeleton, and it is in the form of plates called sclerites, targa, sterna, pleura. These are joined by flexible membrane or chorial membrane, allowing the movements. Okay, and head is formed by the fusion of and it lies at right angles to axis of the body, so that mouth parts are directed downward. Such a head is called as hypognathous head. Such a head is called as hypognathous head. And head shape. And in the head region, if you observe, pair of compound eyes, and in front of the compound eyes membranous socket is there, from which arise antenna. Antenna help to monitor the surrounding. Their sensory structures detect the changes happening, and near the base of antenna white spot is present, ocellus or fenestra equal to simple eyes. And then there are mouth parts at the free end. Type of mouth parts: biting and chewing, upper lip or labrum, lower lip or labium, one pair of mandibles. They have incising and grinding regions to grind the food material. These are pair of maxilla. This is lower lip or labium. And there is a hypopharynx, which is equal to type of mouth parts are biting and next. Head is connected to thorax by a narrow flexible neck. Neck is not a separate division, it's anterior extension of the prothorax. Head is capable of moving all around because of this neck only. Okay, next one. Thorax. Thorax is having three segments: prothorax, mesothorax, and targal plates covering these are called pronotum, mesonotum, metanotum. Pronotum is so large, it covers prothorax, neck, partially head also covered up by that. And the thorax, we have two parts, three pairs of legs, each segment bearing one pair of legs, and dorsally two pairs of wings. Which segment is without wings? Four wings are mesothoracic, hind wings are forewings are also called tegmina or elytra, and they can be described as thick, leathery, opaque, meant for protection, not for flight. Hind wings are transparent, thin, membranous, and are meant for flight. That's it about. Next one, abdomen. Segments 10, covered by 10 targa dorsally, but only 9 sterna ventrally. Here they mention only this brood pouch. In female, 7th sternum is boat shaped, along with 8th and 9th sterna form the brood pouch. In the brood pouch, anteregion, female gonopore, spermatical opening, collateral glands are seen opening. lining. Male brood pouch, dorsally bounded by 9 and 10 carga, ventrally by 9 sternum. In the brood pouch, you can see dorsal, anus, ventral, male gonopore, and chitinous structures called as gonapophysis. Other than that, 10th segment bears a pair of segmented structures, anal styles. They are sensory, occur in both the sexes. Anal styles are unjointed structures only in males. That's sexual dimorphic feature of cockroach. Done. Next one. Anatomy. In the digestive system, mainly check that sequence. Mouth followed by no buccal cavity. Short pharynx, then short narrow esophagus. Esophagus dilates to form a large sac-like structure called crop, where food is stored temporarily, and it ends with gizzard, also called as proventriculus. Up to this internal lined by chitinous cuticle. Then midgut. Midgut is not having inner cuticular lining. Then gizzard, also called grinding meat. It has outer layer of circular muscles and inner chitinous layer forming 6 plates which help in grinding the food material. And then junction of foregut midgut 6 to 8 hepatic cecae or mesenteric cecae or gastric cecae are present. Function secrete digestive juice. And then midgut hindgut junction 100 to 150 malpighian tubules. They open actually into ileum and they are excretory structures. Those seen in the gut, nothing to do with the digestive. Hindgut includes 3 parts: short, narrow ileum, long coiled, wider colon, and sac-like rectum. Rectum opens out through anus. The sequence of parts, already 2-3 times they framed a question on the sequence of the parts. And then a pair of salivary glands. Cockroach food. What does it feed? Omnivorous. They are nocturnal, active at night time, omnivorous and cursorial, fast running animals. So salivary glands, 1 pair on either side of the crop region. That's all about it. What is the next one? Blood vascular system. Blood vascular system is open type. No blood vessels? Poorly developed. Actually, the anterior aorta is considered as blood vessel only, and here all tissues, organs are bathed in the blood. Such a blood is called hemolymph. Blood is colorless. It has colorless plasma, colorless cells called hemocytes. Everything is colorless, no pigment. And body cavity is divided into sinuses: pericardial, perivisceral, perineural sinus. Heart is long tubular, 13 chambered, runs along the side of thorax and abdomen. So 3 plus 10, 13 segmented, and each chamber is funnel shaped. Paid openings lateral side, ostia. Paid openings are called ostia, and the blood direction of flow is posterior to anterior. Lateral openings are ostia. And anteriorly heart continues as anterior aorta opening into the head region. Regarding circulation, from the sinuses, blood enters into heart chambers through ostia, continues to flow anteriorly through the aorta into the sinuses. Again from the sinuses return. That's how circulation occurs. Which muscles help in circulation? Alveolar muscles. And the next one, respiratory system, tracheal system. Blood cannot transport oxygen, that's why network of tubes carry the air directly to cells and tissues. It includes 3 components: spiracles or stigmata, trachea, tracheole. 10 pairs of spiracles through which air enters, and trachea divide to form 5 net branches called tracheoles. Main point: exchange of gases occurs at tracheoles by diffusion. Okay, next one. What is the next one? Excretion. Malpighian tubules, 100 to 150, yellow color and present at the junction of midgut and hindgut, lined internally by glandular and they collect nitrogenous waste from the hemolymph, convert it into uric acid are discharged into hindgut. That's it. In addition to that, nephrocytes, uricose glands, fat bodies also help in excretion. Next one, neural system. Neural system or nervous system, it includes brain, supraesophageal ganglia, subesophageal, forming a nerve ring around the esophagus. Only that part is in the head. Major part is along the ventral side of the belly region. That's why even if head is cut off, it can remain alive for 1 week time. And double ventral nerve cord. How many ganglia are there? 9. 3 in the thorax, 6 in the abdominal region. That's it. And sensory structures. Antenna, labial palps, maxillary palps, anal cerci, compound eyes. And each compound eye is made up of how many units? 2000 units. And these, every unit is capable of receiving that means each eye can form little 2000 images. That's why it's called mosaic vision, characterized by more sensitivity but less resolution. It can receive more light but clarity is less. And it is important because they are nocturnal. Nighttime light availability intensity is less, so they need to take more light with the help of this. And then female male reproductive system, testis location 4 to 6 abdominal segments. From each testis arise vas deferens. The two vasa differentia open through seminal vesicles into ejaculatory duct, which opens to the outside. So sequence: testis, vas deferens, seminal vesicle, ejaculatory duct. Sperms are stored in seminal vesicle, glued together to form bundles called spermatophores in seminal vesicles only. Okay, and other than 6th and 7th segment, there is a mushroom shaped gland. It has two components: peripheral longer tubules and short central tubules. Utriculi majoris, utriculi brevis. Nourishment, and it forms a coating of the spermatophore. Plus another gland is there called phallic gland or conglobate gland. When spermatophore comes out, how many layers are there? 3. Inner layer produced by utricular mushroom gland, second layer by ejaculatory duct, third layer by secretion of conglobate or phallic gland. And there are 3 chitinous structures called gonapophysis, also called phallomeres, right, left and ventral. Left one is associated with the pseudopenis and titillator. They help in copulation. And then female reproductive system. It includes pair of ovaries in 2 to 6 abdominal segment. Each ovary has 8 ovarioles in which ova are seen in different stages of development. And from each ovary duct arises called oviduct. Both the oviducts join to form common oviduct or vagina, which opens into brood pouch through female gonopore. And there is one pair of spermatheca, right, filamentous left sac-like, which receives and stores the sperms after copulation. And a pair of collateral glands, their secretion forms egg case or ootheca. These are the components. 3 pairs of gonapophysis are also there. And what happens during copulation? Spermatophore is transferred to the brood pouch of the female, and all the sperms of that get transferred to spermatheca and stored there. So sperms are stored in males or before copulation in seminal vesicle, after copulation or in female by spermatheca. And once sperms, ova are formed, how many ova can be released? 8 from each. So 16 ova released into the brood pouch through female gonopore. Immediately spermatheca also discharges the stored sperms. So fertilization occurs there. That's why fertilization is said to be internal. And immediately collateral gland releases secretion on them, which hardens to form ootheca or brood pouch. So not brood pouch, egg case, in which these eggs are arranged in 2 rows. How many ootheca are formed? 9 to 10. Each ootheca, how many young ones got? 14 to 16. These are, again, size, color is mentioned. They are deposited at places where food is available once young ones come out. Young ones which hatch out look like adults and are called as nymphs. They undergo how many moltings? 30. And such a gradual development is called parometabolism. Next. Next. Economic importance, nothing. They actually spread disease, contaminate the food and all that. Frog, Indian bullfrog, Rana tigrina, and it belongs to phylum Chordata class, amphibia, order Anura, and these are coagulotherms. That means body temperature changes along with surroundings. That's why it is difficult for them to survive during extreme summer and winter seasons. Undergo estivation and hibernation. Summer sleep and winter sleep. During these periods, they go down to the bottom, but make burrow, live inside that, and all activities are slowed down just to keep it alive. Minimal activities occur, stops feeding and use only reserve food. Only cutaneous respiration occurs at that time. That is about it. And they also have the ability to change their body color as per the surroundings. Camouflage or mimicry to protect from enemies or predators. And then regarding morphology, body is covered with two parts, head and tongue, neck absent, tail absent. Skin is soft, slimy, moist. No skeleton, scales are absent. Dorsal olive green with irregular spots. Yellow, pale yellow, uniform ventral side. And it absorbs water through the skin. Does not drink water. That's why they are not seen in marine habitats also. They can live only in fresh water. They die in sea water because water moves to the skin. And coming to the other points. Wide mouth, above that pair of nostrils, and then bulged eyes covered by upper and lower eyelid. Mobile eyelid is lower, in the water protected by nictitating, and behind the eye a circular patch of skin tympanum to receive the sound vibrations. Trunk region, just two pairs of limbs. Four limbs short, each with 4 digits. Thumb is absent, and in males it bears copulatory pad. And hind limbs each long, folded with 5 digits, and they have webbed feet. Web helps in swimming. Other sexual dimorphic feature, vocal sacs. Vocal sacs and copulatory pads occur only in males, absent in females. In the body cavity, you see all the systems. Regarding digestive system, mouth bounded by upper and lower jaws. Mouth leads into buccal cavity followed by pharynx. And here the points are, upper jaw has teeth, lower jaw and tongue is bifid, attached in front, free behind, reversible, helps in capturing these are the main points. And then it leads into short esophagus followed by stomach, intestine, rectum, opens into cloaca through anal opening. And in this case the other points are, intestine is short. Why? Carnivorous condition. And digestive glands, salivary glands are absent. Largest gland, liver, secretes bile juice. Bile juice contains bile pigments, bile salts, but no enzyme. Bile salts help in emulsification of fats. Pancreas produces pancreatic juice containing all the enzymes. So only the salivary glands are absent in the case of frog. So tongue helps in capturing the prey. No digestion occurs here because no salivary glands, no mastication also. Simply prey is swallowed. Digestion begins after reaching stomach. Gastric glands help in that, and after that bile, pancreatic juice and all that. In the intestine, villi and microvilli are present, which increase the area of ulcer. Plus bile juice, pancreatic juice, both carried by common duct into duodenum. They are the main points. Okay. Next one. What is chyme? Partially digested, every food formed in the stomach is called as chyme. And then coming to the respiration, 3 types of respiratory structures. In water only, skin. On land, skin, buccal cavity, lungs, pink structures, and the anterior part of the, just give one reading. Okay, mainly which respiration occurs, which place, that is more important. Hibernation, respiration also, only cutaneous respiration. And regarding circulatory system of heart, already we discussed 3 chamber, 2 atria, 1 ventricle. On dorsal side there is a triangular structure called sinus venosus, receives 2 precavals and 1 postcaval or vena cava, and this opens into Right atrium. Left receives from lungs. And ventricle pumps the blood through a conical structure called conus arteriosus, which supplies blood through arches, arterial system. So if you take circulation, ventricle, conus arteriosus, arteries, arterial system to all body parts, from body parts collected by veins, vena cava, sinus venosus opens into right atrium. That is the sequence. Sinus venosus is on dorsal side, conus arteriosus is on ventral side. Triangular structure is sinus venosus, conical structure is conus arteriosus. That's enough. And in addition to that, the special points are blood dust, all everything is same. RBC are nucleated. That's a special point. Lymphatic system is also present. And another thing, both hepatic portal system and hepatic, you know anyway, renal portal system carries the blood from lower parts of the limbs to kidneys. Okay, that is the one which is to be highlighted. And then the next one, excretory system. Pair of kidneys and what are the structural function units in the kidney? Nephrons or uriniferous tubule. Excretory product is media, and it is the two ureters open into cloaca, and there is a bilobed urinary bladder opening into cloaca. It lies ventral to the rectum. That is where urine is temporarily stored. And one more important thing, in males, ureters are also called urinogenital ducts because both are transmitted through the ureters. And then coordination, chemical coordination, neural coordination, both are there. Chemical coordination, nothing is there, just names of the glands, pineal, pituitary and all that, secreting hormones. Regarding neural, brain, three parts, forebrain, midbrain, hindbrain. Two meninges, uromater and pyometer. Forebrain includes pair of olfactory lobes, pair of cerebral hemispheres, unpaired diencephalon. Midbrain includes pair of optic lobes, corpora, bigemina. Hindbrain includes cerebellum and no pons, nothing. Simplified structure, only cerebellum and medulla. And medulla emerges out of cranium through a large opening, foramen magnum, to continue as spinal cord. Number of cranial nerves, 10 pairs. Spinal nerves, 9 or 10 pairs. In the case of frog. Okay, that's it. And sensory structures, all 5 senses are there, but only eyes and ears are well organized organs. Ear not only for hearing but also for balance. Remaining 3, tactile in the sensory papilla of the skin, olfactory and gustatory, taste receptors on the tongue. These are not well organized. They are aggregations around the nerve endings. That's it. Not organized into organs. Last one, reproductive system. This is the male reproductive system. Testes are oval, yellow colored, present at the upper end of the kidney, attached by double fold called mesoarchaeum. And inside the testis, seminiferous tubules produce the sperms. They are carried out by vasa. In cockroach, vas different. No different. Humans both are there. Okay. Vasa differentia emerge out 10 to 12. They enter the kidney and inside the kidney open into Bidder's canal. Bidder's canal only for sperm transport, not urine. And then Bidder's canal through transverse canals opens into ureter. Ureter transports urine as well as the sperms and acts as urinogenital duct, which opens into cloaca. Cloaca opens out through cloacal aperture. Done? That's it. That means male reproductive system structurally and functionally associated with the kidneys, because vasa efferentia entering into the kidneys, opening into Bidder's canal. Bidder's canal occur in kidney, not in testis. Female reproductive system. In the female reproductive system, how many ovaries? Again, one pair, and ovaries are structurally related. That means they lie close to the kidneys, attached by mesovaria, but no functional relation, because a pair of separate oviducts are present for the transport of ova. That means in females, ureter should be called ureter only. You shouldn't use the word urinogenital. That is applicable strictly only to males, because separate ducts are there. and this is the ovary. Once ova produced, they are released into the coelom. From the coelom, they are collected by these oviducal funnels. Opening is called ostia, and these oviducts transport them into the cloaca. Before opening into cloaca, it is slightly enlarged where eggs are temporarily stored, called as ovisa. So ova transported like this and released into cloaca, discharged out. That is a description. These are fat bodies found in both. Last point, breeding season is rainy season. During the breeding season, males try to attract the female by producing loud croaking sound with the help of vocal sacs. When female comes closer, male mounts on the back of the female and holds it tightly with the help of amplexatory pads, because skin is slimy or slippery. So there will be no grip, that's why they use these pads. And both the animals release their gametes into water. That means it's not a true act of copulation, because in copulation sperms must be discharged into female tract. But here it's not happening, both are releasing gametes. That's why it's pseudo copulation or amplexus. So what is the site of fertilization? Water. Internal or external? External. Development also external. Development is indirect because there is a larva. How many eggs are released at a time? 2,500 to 3,000. What is the larva called? Tadpole larva. Tadpole resembles fish. All aquatic adaptations are seen: gill breathing, herbivorous, fins help in locomotion. Such a fish-like larva undergoes metamorphosis to become limbed, lung-breathing, carnivorous, almost all transition, adult stage, and the hormone required for this is thyroxine hormone. And not only that, frogs are having economic importance also. They feed on insects, thereby control insect population, help the farmer. They are important links in food chains and food web, maintain ecological balance, and legs are muscular and taken as food material. Let us wind up now, okay? In cockroach, location of mushroom gland 6 and 7. Testis 4 to 6. Ovaries, spermatheca 6. Usually they frame questions on this matching type questions. Spermatheca is in the 6th one. Okay, now coming to the next topic, human reproduction. I think second year syllabus is much fresh in your mind. First year syllabus. What are the events that occur at the age of puberty? Actually, humans are viviparous and sexually reproducing. Viviparity, giving birth to young ones, and the events are: first step, formation of gametes called as transfer of semen into female reproductive tract, leads to fertilization. Development of zygote into embryo, blastocyst. Blastocyst attaching to uterine endometrium. That results in pregnancy or gestation. That finally ends with parturition or childbirth. So all the events related to reproduction, they start taking place only from the age of puberty, and there are significant differences between males and females. Gametogenesis continues even in the old age in the case of male, but stops around 50 years, called as menopause in the case of. Now coming to the description of male reproductive system. Male or female reproductive systems, they are located in the pelvic region. Pelvic region means lower abdomen, thorax, abdomen, separated by diaphragm. Upper abdominal cavity, lower abdominal cavity. Lower abdominal cavity is supported by pelvic girdle. So this is also called as pelvis region. So male or female systems are located in the pelvis region. And reproductive systems, male or female, they include primary sex organs, nothing but gonads, testes and ovaries. They produce gametes and as well as produce the hormones. Other than that, accessory ducts, accessory glands occur. Ducts for transport and glands to secrete the secretions related to the reproductive system. Okay. And in addition to that, external genitalia, which are visible outside the body. These are the components that we are going to discuss. And in male reproductive system, testes actually develop in the lower abdominal cavity during the fetal stage, but descends into extra abdominal pouch called scrotum just before the birth. And why is it necessary? Because the temperature required for spermatogenesis is 2 to 2.5 degrees less than body temperature. If they do not descend, they will be exposed to abdominal body temperature, which becomes too high for spermatogenesis. Failure of testis to descend, it is called as cryptorchidism. And coming to other points, during the descent from the pelvic region, testis descend passing through a canal called inguinal canal and remains lodged in the scrotum, and it remains attached to wall of scrotum by gubernaculum, remains attached to abdominal wall by a spermatic cord, which includes blood vessels, nerves, vas deferens, cremaster muscle. Cremaster muscle help to regulate temperature within the scrotum, maintains the temperature within the scrotum. Not only cremaster muscle, Dartos muscle in the wall of scrotum also help in regulating the temperature within the scrotum. And then each testis, dimensions given 4 to 5 centimeters length, 2 to 3 centimeters, and it is oval shaped structure. This is covered by a dense fibrous covering, tunica albuginea. Outside that, double-layered fluid-filled tunica vaginalis. Inner to that, highly vascularis is present. Main layer, tunica albuginea, dense fibrous layer, connective tissue layer. It invaginates to form septa, dividing the interior into 250 compartments. They are called testicular lobules. In each lobule, how many seminiferous tubules are there? 1, 2, 3. So totally 250 to 750 seminiferous tubules in one testis. One testis. Okay. And that is a number. And each seminiferous tubule is lined by two types of cells. What are they called? Germ cells and these are the tubules in transverse section, lined by germ cells and Sertoli cells. Germ cells give rise to primary spermatocytes, and then they undergo meiosis to form sperms. So actual spermatogenesis occurs from the germ cells. Between them there are tall cells called Sertoli cells, nurse cells or sus-tentacular cells. They provide nourishment to the developing gametes, that is germ cells or developing spermatocytes. And outside or in between the tubules, interstitium is present, in which you can see interstitial cells called Leydig cells, which secrete androgens, and in addition to that, blood vessels and immunocompetent cells are also present in the interstitium. That is the internal structure of testis. All these seminiferous tubules, they lead into fine ducts called as retitestis. So this all these are seminiferous tubules. These seminiferous tubules, all of them lead into a network of tubules called as retitestis. Retitestis lies inside the testis only, near the margin or border, and all these join to form fine ducts called vasa efferentia. So seminiferous tubules lead into retitestis, retitestis leads into vasa efferentia. Which ones leave the testis? Vasa efferentia, and they open into highly convoluted tubular portion called as epididymis. Epididymis is again differentiated into caput epididymis, corpus epididymis, cauda epididymis. Head, body, tail, equivalent regions. And sperms are stored for some time in the epididymis and undergo maturation there. Okay, and then this lies posterior surface of the, this is epididymis, lies along posterior surface of the testis. And the last part of epididymis, cauda, continues as vas deferens. It takes a turn, runs back, ascends into the abdominal cavity, and in the abdominal cavity, it loops around the urinary bladder. Is this part clear? Now the question, part accessory, what is the first accessory duct? Retitestis. Accessory ducts enclosed within the testis. Retitestis. Accessory ducts which leave the testis. Vasa efferentia. Which leave the scrotum. Vas deferens. Because this is a scrotum, right? Leaving the scrotum is vas deferens. And then the next part, this diagram, this is very clear. See, this is the epididymis, vas deferens, enters the abdominal cavity, takes a loop around urinary bladder, then this is seminal vesicle. The end of the vas deferens joins a duct coming from seminal vesicle to form ejaculatory duct. Ejaculatory duct opens into urethra in the middle of prostate. This red color one is, pink one is prostate gland, in the middle of the prostate gland. And this duct is urethra, which actually starts from urinary bladder. Its main job is to transport urine towards the exterior, but ejaculatory ducts are discharging the sperms into that duct. That's why it acts as a shared or common passage for transport of urine as well as sperms. This is very long. It can be differentiated again into part enclosed within the prostate is prostatic urethra. This part not enclosed by any structure is membranous urethra, and this portion done into the penis is called as penile urethra, longest being. Next, that is passage, and it opens at the tip of the penis. Then coming to the other structures, male accessory glands include pair of seminal vesicles, one prostate gland, pair of bulbourethral glands. And the secretions of these glands constitute seminal fluid or seminal plasma. 60% of that contributed by seminal vesicles. Prostate gland 15 to 30%. Unpaid gland is prostate. Remaining two are paid glands. And the secretions are rich in fructose, calcium, certain enzymes. One more thing, secretion of bulbourethral gland helps in lubrication of the penis, and they are also called as Cowper's glands. That is about the gland associated with that. Now coming to external genitalia, scrotum and penis, externally visible parts, so they come under external genitalia. And scrotum is made up of special erectile tissue, and it makes penis erect, which helps in insemination. With erection only, it can easily transfer the semen into or discharge the semen into the female reproductive tract. And if you consider the structure, this is made up of literally 3 columns of tissue, 2 dorsal columns, carpora cavernosa, 1 ventral column called as carpus spongiosa, and urethra actually runs through the carpus spongiosa. So it is also called spongious urethra also, because it runs through the spongiosa. And another thing, the terminal part of the penis is enlarged or bulbous in nature, which is called as glans penis, and the loose fold of skin covering that is called as foreskin or prepuce. Next one. How many ejaculatory ducts are there? One pair. We discussed one side system only. Okay, next one is female reproductive tract. And in the female reproductive system, primary organs are ovaries. They are located in the pelvis region, that is lower abdomen, and they are present on either side, 2 to 4 centimeters in size, and ovary remains attached to uterus as well as pelvic wall, this abdominal wall, by ligaments. Uterine ligament, suspensory ligament. So this is the system. These are the pair of ovaries attached to uterus as well as the abdominal wall. And then ovary is covered by germinal epithelium or thin epithelium, from which germ cells are actually produced, and it encloses stroma. Interior of the ovary is called stroma. Stroma is differentiated into outer cortex and inner medulla. In the cortex, you can see a number of follicles in different stages of development. Medulla is made up of loose connective tissue. So that is about the ovary. And accessory ducts include pair of fallopian tubes, uterus, cervix, vagina. All these structures along with the mammary glands work together to support various functions, general statement. So fallopian tubes, also called oviducts. One pair on either side start from the periphery of the ovary and extend towards the uterus. Length of the duct internally lined by ciliated columnar epithelium, and it starts with a funnel shaped structure. Infundibulum. Margin of infundibulum forms finger-like process. Fimbriae, meant for correction of the ova released during ovulation. Infundibulum leads into ampulla, which is having wider lumen, plus it is a site of fertilization that leads into last part called isthmus. Isthmus has a narrow lumen. Isthmus opens into or joins the uterus. So last part of the oviduct, isthmus. Closest to the uterus, closest to the ovary. First part is, and it is attached to abdominal wall by mesosalpinx, ovary by mesovaria. Both the oviducts open into uterus, which is median in position, also called womb, metra, hystera. Womb, metra or hystera. Inverted pear-shaped structure. Thick-walled muscular and highly vascular structure. And in the uterus, the elevated dome-shaped part can be called as uterine fundus. This is the body. Lower portion is the cervix. Wall of the uterus has three layers. Outer thin membranous, middle thick wall, made up of smooth muscles, inner glandular. Endometrium undergoes periodic changes during menstrual cycles, and myometrium is responsible for contractions during the childhood. So that is about uterus. And the lower portion, cervix, lumen of the cervix and vagina together constitute birth canal. Vagina opens to exterior directly through vaginal orifice. Okay, that is a description. Accessory glands. Coming to the glands, female accessory glands are two types, Bartholin glands and Skene's glands. Bartholin glands are homologous to. Skene's glands are homologous to prostate gland. Skene's glands are also called paraurethral glands. And in addition to that, external genitalia of the female system. Remember the parts? It includes mass of fatty tissue covered with skin and pubic hair called as mons pubis. From the mons pubis, a pair of fleshy folds extend downwards surrounding the vaginal opening called labia majora. Another small pair under the majora is called labia minora, and the two openings seen at the center are upper urethral opening, lower vaginal opening. Surrounding these two only these fleshy folds are there, and upper junction of labia minora just above urethral orifice. There is a tiny finger-like structure called clitoris made up of erectile tissue and homologous to pens. And vaginal opening is covered partially by a mucous membrane called as hymen. Hymen usually torn during first coitus or intercourse, but not, that is normally, but not necessarily. Sometimes it gets torn or breaks down due to other activities. Sudden fall or jolt which causes impact in the pelvic region, usage of vaginal tampons, or taking part in activities cycling, horseback riding, which involve vigorous movements in the pelvic region can cause, that means due to this vigorous motility, sometimes due to the impact, it can simply rupture. Plus, another exception, in some women, after taking part in sexual intercourse also, it remains persistent. All these conclude that presence or absence of is not a reliable indicator of virginity. Done? Next one. Mammary glands. Mammary glands are one pair. These are the modified sweat glands. modified sweat glands, and in the case functional glands are characteristic of females, and in humans one pair located in the thoracic or chest region, and each breast or mammary gland is made up of, it's a gland, naturally glandular tissue. In addition, variable amount of fat tissue is also present. So made up of glandular tissue and fat tissue. And inside each mammary gland, there are 15 to 20 mammary lobes. Each lobe contains many clusters of cells called alveoli. Alveolar cells secrete the milk stored in alveolar cavity. From the cavity, milk is carried out, passing through mammary tubules. All mammary tubules of each lobe join to form mammary ducts join to form with wider lumen mammary ampulla, and they open through lactiferous duct at the nipple. Lactiferous ducts open at the nipple through which milk is suckled by the baby, and the pigmented region surrounding that is called as areola. Done? Next one. Gametogenesis. If you consider the gametogenesis process, there are again considered, gametogenesis is formation of gametes. It is called spermatogenesis in male and oogenesis in female. There are remarkable differences between the two, and you know this. Just to follow the labeling as given in the textbook. Okay. In males, spermatogenesis starts at the age of from the puberty only it starts, and germ cells which form the lining of seminiferous, spermatogenesis occurs inside the seminiferous tubule. Germ cells multiply by mitosis. They are also called spermatogonia or sperm mother cells. They simply multiply by mitosis to increase in number, and after many divisions, when number is sufficient, they stop dividing and differentiate to become primary spermatocyte. They use different colors for different stages. This is primary spermatocyte. All these are spermatogonia. Okay, just increase in number. And then spermatogonia, primary spermatocyte, both are diploid with 23 pairs or 46 chromosomes. Now the primary spermatocyte undergoes first meiotic division, and you know that it's a reduction phase. So each primary spermatocyte divides equally because the daughter cells are equal size, and these two are haploid because this is the reduction division. And these are called secondary spermatocytes. So one primary spermatocyte gives two secondary spermatocytes. Each secondary spermatocyte completes second meiotic division to form totally four spermatids, and each spermatid undergoes differentiation to become spermatozoa. This process of conversion of spermatid to spermatozoa is called spermiogenesis. It does not include any division. It is simply differentiation, undergoing structural changes. That's it. It's called differentiation. Clear? How many spermatids will be formed from 200 primary spermatocytes? Huh? How many spermatozoa? From 200 spermatids. 200 spermatids. 200 spermatozoa. For 200 spermatid formation, how many primary spermatocytes are required? Huh? Secondary spermatocytes. Spermatids. One primary spermatocyte forms how many sperms? How many spermatids? One secondary spermatocyte forms how many spermatids? You know that. Have clarity? Okay. That is about it. And spermatogonia, primary spermatocytes are Diploid. All other stages are haploid. What is the first haploid stage? Second is haploid. That's it. Okay. And then hormonal control. Spermatogenesis begins at the age of puberty. At the age of puberty, hypothalamic hormone GnRH level increases considerably. That stimulates anterior pituitary to secrete gonadotropins, FSH and target cells for FSH. They produce factors like androgen binding protein, which helps in spermiogenesis. Plus, Sertoli cells also secrete inhibin, which inhibits the secretion of FSH. Inhibin inhibits the secretion of FSH. And LH target cells, Leydig cells, they secrete androgen. Principal one is testosterone. And androgens are required for spermatogenesis, and already you have learnt all the secondary sexual characters and all that. That can be repeated again. Now coming to the next step, sperm structure. Spermatid differentiates to become spermatozoa, and a sperm is having four parts: head, neck, middle piece and tail regions. It is a haploid cell. Whole sperm is covered or enveloped by plasma membrane. Head is nothing but elongated condensed nucleus, haploid in nature. And anterior to the head, there is a cap-like structure called derived from Golgi complex, contains sperm lysins. That means enzymes which help to penetrate into the egg at the time of fertilization. These are the enzymes of the sperm, so sperm lysins. And neck region, it has two centrioles, proximal and distal, perpendicular to each other. Distal centriole gives rise to axial filament. Axoneme or axial filament runs through the middle piece and continues as a tail region. And middle piece, it has many mitochondria arranged spirally around the axial filament. Arranged spirally around the axial filament, and that spiral sheath of mitochondria is called Nebenker. Mitochondria provide the energy required for motility of the sperm. Last portion is tail. Tail is nothing but axial filament covered by plasma membrane. Whole axial filament is covered except the tip part where it is naked, and tail function is causing motility. So, sperm is able to swim or show motility due to tail. Energy for that is provided by mitochondria. That is the structure. And again, conclusion part. During coitus, the ejaculate release discharge contains 200 to million sperms, of which for normal fertility, minimum 60% must be having normal shape and size, of which vigorous motility. Then only there is scope for fertility. So if you consider normal shape, normal size, vigorous motility, combination of all the three, it should be 24% of the total sperms. 40% of 60%. 24% of the total sperm count. 48 to 72 million. And not only that, secretions of epididymis, vas deferens, seminal vesicle, prostate gland, essential for maturation and motility. Not only the glands, even epididymis and vas deferens secretions also. Sperms with this seminal plasma or fluid together constitute semen, and the functions of accessory ducts and glands maintained by androgens produced by the testes, that is Leydig cells. Next comes oogenesis. Oogenesis is initiated in the early embryonic stage or fetal stage itself. Initially, in each ovary, a couple of million oogonia are formed. That means, look at this, again the same points are given. Germ cells undergo mitosis, that is number increases, and then differentiate to primary. Same structure, same procedure. The only thing, The oogonia formation, that means up to here, mitotic division. After that, no division. That means oogonia cannot be added later stages. Here, these cells keep on dividing. There are stem cells which can divide continuously. So, in the fetal stage, a couple of million oogonia are developed in each ovary. Afterwards, there is no increase in the number of oogonia. And then, oogonia become primary oocyte and start meiosis 1, but the division is arrested at prophase 1. This happens in fetal stage. Now each primary oocyte gets surrounded by single layer of granulosa cells. It is called as primary follicles. So up to the formation of primary follicles, it happens in the fetal stage. Each primary follicle has primary oocyte surrounded by single layer of granulosa cells. No further change occurs till the age of puberty. This happens in fetal stage. Afterwards, birth to puberty, no change occurs. Moreover, many primary follicles degenerate from birth to puberty, leaving only 60 to 80,000 in each ovary. That means out of 2 million, only 60 to 80 are left in each ovary. They only continue further process after puberty. Done? Next. Puberty onwards, menstrual cycles begin. Each menstrual cycle, few primary follicles begin to undergo further development, but only one would be able to complete the development, that is a dominant follicle, and that releases the ovum. Other follicles meanwhile degenerate again. That's what happens during the procedure. So, first step when development begins is primary follicle soon changes to. What is the difference? Primary oocyte now surrounded by many layers of granulosa cells and get covered by theca. That is second. Many layers of granulosa cells, theca. That is characteristic of secondary follicle. Primary oocyte does not undergo any change. Changes occur only follicle. And later it develops into tertiary follicle. Tertiary follicle means there develops a fluid filled cavity called antrum, and not only that, theca differentiates to form theca externa and theca interna. Externa is fibrous layer, interna is cellular layer. Interna and granulosa cells, they secrete estrogen. Growing follicles secrete estrogen that comes from the granulosa cells and inner theca interna. Okay, and what is the stage called? Tertiary follicle. In this stage, primary oocyte grows in size and stimulated to complete meiosis 1, which is unequal division, forming a large cell called secondary oocyte and a tiny cell called first polar body. Both of them are haploid with, because it's a reduction division. And secondary oocyte retains bulk of the cytoplasm of primary oocyte. And secondary oocyte starts meiosis 2, but the division is again arrested in the metaphase 2. Okay, soon tertiary follicle develops into mature follicle called as Graafian follicle, and you can see a glycoprotein layer surrounding the secondary oocyte. What is that called? Zona pellucida. Innermost layer of granulosa cells, which are firmly attached to zona pellucida, constitute corona radiata. Non-cellular layer surrounding the oocyte, acellular, non-cellular, or simply glycoprotein layer, zona pellucida. Cellular layer is corona radiata. What is the stage of the oocyte? Secondary oocyte. Further development cannot occur. Now it is ready to undergo ovulation. That means that mature Graafian follicle ruptures, releases the secondary oocyte with its coverings, zona and corona radiata. That process is called ovulation. What is the stage released? Actually, in the new edition, in the diagram, they made the change. So now, no controversy. Actually, that's the controversial point because theory says secondary oocyte and it is labeled as ovum. Actually, it is secondary oocyte. They made the change, correction is done. So, secondary oocyte is the one which is released at the time of ovulation. So, which is the stage released during ovulation? It would be immediately collected by fimbriate and transported to that is ampullary region of the fallopian tube. That means it is still oogenesis is not yet completed. Now it will be complete in the fallopian tube, the last step in the fallopian tube. And before continuing, let us complete this one. After the ovulation, the remnants, remaining parts of the ruptured follicle, they proliferate, divide, develop yellow pigment. Carotene. Carotene pigment is developed. Such cells with carotene are called lutein cells and it is called corpus luteum or yellow body. This is temporary endocrine structure and it secretes the hormone mainly progesterone and some amount of estrogen also. And it secretes even inhibin also. Granulosa cells also secrete inhibin. This also secretes inhibin. Males inhibin is produced by Sertoli cells. In females by granulosa cells or corpus luteum. Next, if the released oocyte undergoes fertilization, then corpus luteum will be maintained for next 3 to 4 months, supporting the pregnancy. If fertilization does not occur, it simply degenerates and becomes a white scar called corpus albicans. Degenerates. Now coming back to the completion of the division. Secondary oocyte, it completes the division at the time of fertilization when sperm penetrates into the secondary oocyte. That entry stimulates the oocyte to complete meiosis II, which is again unequal division forming large cell ooted or ovum. Ooted or ovum. No difference because there is no need to undergo differentiation. If you have to arrange in sequence, which one do you write first, ooted or ovum? Corresponding to that, spermatid, ooted. Spermatozoa, ovum. That means ooted actually has to become ovum, but there is no need to undergo any change. So it is simply ooted or ovum. Referred as. And tiny cell is called as second polar body. Whether the first polar body completes meiosis II or not. Okay. Anyway, at the end, polar bodies degenerate. There is no significance. And by the way, this completion of meiosis II occurs with the penetration of the sperm, which happens in the ampullary region of the. That means oogenesis process begins in the ovary and ends in the fallopian tube. Spermatogenesis begins and gets completed within the seminiferous tubule only. And list out the differences. Oogenesis is initiated fetal state. Spermatogenesis, puberty. These cells, they keep on multiplying continuously even in the old age. But oogonia formation never occurs, it stops in the fetal stage itself. Okay, continuous multiplication or division is not observed. Next, beyond early childhood. HbA, HbS, carriers, heterozygous, normally they are not affected. They lead normal life because they show codominance. HbA and HbS exhibit. That means in the heterozygous persons, 50% hemoglobin is normal, 50% is mutated. As 50% hemoglobin is normal, the RBC shape is maintained normal circular biconcave only. When they are subjected to low oxygen condition, then the mutated hemoglobin molecule undergoes polymerization and due to that circular shape changes to sickle cell. So they develop the symptoms only when subjected to low oxygen tension. Talking about heterozygous persons, carriers. And one more thing, a carrier person, what is the probability to transfer the gene to child? 50%. When both the parents are carriers, what is the probability of the child to be affected by disease? 25%. 1:2:1 ratio or 3:1 ratio, phenotypic ratio. So 25% will be affected. Next one, phenylketonuria. It's autosomal recessive metabolic disorder. PAH, phenylalanine hydroxylase enzyme, is not produced because of defective gene. Phenylalanine cannot be converted into tyrosine, so that phenylalanine accumulates and converts into pyruvic acid and all that. Excess level starts appearing in the urine. That's why phenylketonuria. Accumulation in the brain causes mental retardation and hypopigmentation. Skin and hair pigment also decreases. Sickle cell anemia, phenylketonuria, both are examples for pleiotropy, where one gene affects many characters. Both are examples for pleiotropy. And then thalassemia. Thalassemia is again blood related disease, autosomal recessive, and it is marked by reduced production of globin chains. Could be alpha or beta. So we have two types, alpha thalassemia and beta thalassemia. Alpha genes coded by two closely linked genes, HBA1, HBA2. Totally each gene 2 alleles would be there, 4 copies. If one or more mutated or deleted, production of alpha chain. More number of genes affected, less is the production of alpha chain. When alpha chains are less produced, abnormal hemoglobin is formed, which causes easy rupture of the cells, leading to severe anemia. Same manner, beta thal. Where is the gene located? Alpha 16, beta 11. Only one gene for beta, HBB. Okay, same manner. If mutation occurs, there also beta chain production decreases, and that results in again abnormal hemoglobin, leading to anemia. And one conclusion point is quantitative thalassemia, because here the production of number of globin chains is affected. Qualitative, their incorrectly functioning globin chain is produced. Not the problem with number. Modified, altered, mutated chains are being formed, incorrectly functioning. Next one, chromosomal disorders. Chromosomal disorders are mostly caused due to aneuploidy and polyploidy. That is, during anaphase, the chromosomes fail to segregate, remain together. Then one daughter cell receives extra copy, the other daughter cell receives one copy less. So to the normal diploid number, there is addition or loss of 1 or 2 chromosomes, which is a condition called 46 plus 1 trisomy. 46 minus 1, 1 chromosome less copy. And failure of cytokinesis to occur after nuclear division. No cytokinesis, both the sets remain the same cell and there is double of the chromosome number. So it leads to polyploidy. Polyploidy is common in plants, we don't observe in animals. And some of the example, this can occur autosomal or allosomal. Autosomal trisomy, there is extra copy of chromosome at 21st pair, due to which syndromes, many symptoms occur. Down syndrome, after the name of Langdon Down. Almost all are after the names of discoverers only. And short stature, flat round face, flat back, partially open mouth, long wrinkled or grooved tongue, then palm crease, finger loops, congenital heart diseases, physical, psychomotor, that is development also retarded. Mongolism, Mongolian idiocy. Next one, allosomal trisomy, Klinefelter syndrome. Klinefelter syndrome condition is XXY, sex of the person, male. With Y chromosome they are males, but they have extra copy of X chromosome. They are tall stature and gonads are not properly developed, they are sterile, IQ levels also less, and they exhibit some feminine characters like gynecomastia, breast development. And Turner syndrome is, how do you describe? Allosomal, one chromosome is less. They have only one copy of X chromosome and they are short stature. Gonads are not well developed. They are also sterile. Secondary sexual characters also affected. Menstrual cycles do not occur. Breast development does not occur properly. So all those changes occur because without gonads, hormones are not produced. Hormones are responsible for all the secondary estrogen, secondary sexual characters. And female without bar body, Turner female. Male with one bar body. What is the number of autosomes in Down syndrome? 40? 45? 21 is autosome. 45 plus 2 allosomes. Number of autosomes in Klinefelter? 44. Allosomes are 3. In both the cases, total number 47. But number of autosomes, allosomes show variation. So when you are reading the statements, see properly whether they are saying about chromosome, autosome, allosome, because they are all different. Next one. DNA. Human Genome Project started in the year actually with genetic engineering procedures available and sequencing of DNA also possible. They started with all this. They started this prestigious project, Human Genome Project, in 1990. It's called mega project because of the enormous data that is needed. What is the number of nucleotides? Genome? 3. That means 3 billion nucleotides. And what is the cost estimated? Each base pair $3. Total $9 billion. Plus to store the data, how many books are needed? 3,000. So you get the enormous figures or numbers. So it's a very huge project, mega project. And to store this data in typed form or written form, not practically possible. It necessitated usage of computer programs. So along with HGP, other branch developed very closely called bioinformatics. And goals of the HGP to find out 25,000 genes, sequencing the whole 3 billion base pairs, storing the data in databases, improving the tools for data analysis, transfer the related technologies to other sectors, and addressing ethical, legal, social issues that may arise with the project. These are the goals. And then the next one. Actually, this Human Genome Project not only involves determining the human genome, along with that some non-human animals, organisms genome also was studied because it helps us to understand their capabilities and we can use their abilities in fields of agriculture, health, energy production, isn't it? So various other fields also. Some examples are given. Bacteria, E. coli, yeast, Caenorhabditis elegans, non-pathogenic free-living, nematode, plants like rice and Arabidopsis, Drosophila, etc. were also studied along with this project. And what were the two methodologies used? First one, express sequence tags. That means in this method, only genes which are expressed as RNA are determined, not the whole genome. Only the gene sequences which are expressed as RNA. That's why they are called expressed sequence tags. Second method is sequence annotation. It's a blind approach which includes determining whole genome containing coding and non-coding. Total genome. There are some steps. First one, isolation of DNA. Cutting the DNA into very small fragments, because that time sequences they could determine only short sequences. And then cloning. What were the hosts? Vectors. BAC and YAC. Cloning resulted in amplification. We get many copies. And then using automated sequencers, finding the sequence of each fragment. These sequences work based on the principle developed by Frederick Sanger. Also developed the method for amino acid sequence in proteins. And then after determining the sequence of all fragments, arranging them in the correct order, that is the most difficult task, and they needed to use overlapping regions for that. That too, using overlapping fragments also, we cannot do that, humanly not possible. So they developed computer programs to align all the fragments in the correct sequence. Totally, project was started in. They announced that project is complete in. But actually last chromosome completed in May 2000. Last sequenced chromosome 1. Total number of chromosomes 24. 22 autosomes, X and next one. Genetic mapping, physical mapping. I told you to just remember 2 points. Done based on polymorphism of restriction endonuclease recognition sites, polymorphism of repeat DNA called microsatellite. Based on those two, the mapping was done. And then we have the salient features, total size 3164 points, an exact size. Number of genes 30,000, which is very less than the earlier estimate 82. 1 lakh 40,000, and average gene size 3,000. Largest gene dystrophin present on X chromosome, size is 2.4. 99.9% DNA is same for all humans. Function is not known for 50% of gene, discovered gene. Less than 2% codes for proteins. Most of the DNA, it has repetitive sequences. These sequences do not have coding function, but they help us to understand chromosome structure, dynamics and evolution. And the next one, SNPs. Single nucleotide polymer. Sequences differing just in one nucleotide. They are found at 1.4 million locations. They also help to revolutionize identifying the areas of disease causing plus human, tracing the human history. First chromosome has 2968 genes. Y chromosome has 231 genes. That's the data given there. Next one. Applications. Earlier scientists used to study only few genes at a time, but now with complete information of the genome, we can study all genes, how hundreds of genes and proteins work together in coordination and maintain the orchestra of life. Nothing much to be framing a question on that. And then let us come to this part. DNA. All humans differ only in 0.1%, which is about 3 million base pairs. So every time when you want to compare two persons' DNA to find the difference, you have to compare 3 million base pairs of person A, 3 million base pairs of person B, which is a very difficult, time-taking, expensive process. So to make this thing simple, they devised a technique called DNA fingerprinting, where not the total DNA, not 3 million, they just use some specific regions in the genome called repetitive DNA. That means to perform this DNA fingerprinting, repetitive DNA must be collected, separated. How is it done? Density gradient centrifugation. And repeat DNA forms small peaks called as satellite DNA. Rest of the DNA bulk DNA, which can be easily separated. Satellite DNA further classified based on base composition, length of the repeats and number of repeats into microsatellite and mini satellite. And here some important points are why repetitive DNA is actually used for DNA fingerprinting, because it shows high degree of, so easy to compare, because variation is very high, easy to compare. And some 2 points related to that. In a person, DNA collected from different tissues shows same degree of polymorphism. That's why it is easy or it is applicable for forensic studies, because any sample would serve the purpose. And second point, these DNA polymorphisms are heritable. That's why they can be used for paternity or maternity testing in the case of disputes. Clear? Next, polymorphism is the basis for DNA fingerprinting as well as for genetic mapping. So we need to understand what is this polymorphism. Polymorphisms occur due to mutations. Polymorphism, variation occurs due to mutation. Mutation occurring in germ cells. Somatic cell mutation has no significance because it is not passed on. Mutation occurring in the germ cell, which does not seriously impair ability of the person to reproduce. It will be passed on to and spread in the population because their number goes on increasing, that mutation. And once it reaches more than 0.1%, then it is called 0.01%. DNA poly. That means this mutation must occur in the population with high frequency. What is the limit for that? 0.01. Then it is called DNA polymorphism. And polymorphism need not be non-coding region. It can be in coding region also, which is observable change. When mutation occurs in alleles, we can observe the change. But the chance of observing polymorphism is very high in. Why? Changes occurring in non-coding region cannot show any effect on reproducibility of the person. So definitely the person reproduces and that mutation is passed on to next generation, again to next generation. So they keep on accumulating generation after generation. So polymorphisms, that's why very common in non-coding regions. And these polymorphisms are important because they help in evolution and speciation. Without change, how can new species develop? Changes are polymorphisms. They help in evolution and speciation. Next one. The technique of fingerprinting developed by Alec. And what was the method used by him? Southern blotting. And which probe was used by him? VNTR. What is VNTR? Variable number tandem repeat. The tandem repeat means sequence repeated end to end. The number of repeats vary individual to in one individual from chromosome to same chromosome at the same locus also differ because one is maternal, one is paternal. That's why the number is highly variable and that is used in this. Steps in this first one. Isolation of DNA by using restriction endonucleases. Separation by using gel electrophoresis. Then blotting, transfer of these fragments onto synthetic membranes like nylon or nitrocellulose. And then hybridization using radio-labeled VNTR. Detection of hybridized fragments by using autoradiography. As probe is radio-labeled, wherever probe is hybridized, it exposes the film which gives a pattern of banded appearance which is called DNA fingerprint. Okay, next one, there is a conclusion. VNTR size variable from 0.1 to 20 kb because number of repeats highly variable, size is also high. Is it mini satellite or micro satellite? Yes. So that is the conclusion. Nowadays it is being used in population genetics. And one more point is there. If the sample available for fingerprinting is very limited, can it be done? How? PCR helps in amplifying, then it can be performed with ease. That is another point mentioned there. What else we need to discuss? Evolution. What is the introduction paragraph? Just give one reading, looking at the sky, stars, peeping back. Basically, distance between stars is measured in light years, meaning far located, stellar distances. Let us focus on that. Evolution is study of origin of life and subsequent appearance of many variations or variants in the. To understand origin of life, first we need to understand origin of earth, because life originated on earth. That means what were the conditions which allowed this? To understand that, you need to understand earth, and to understand earth formation you need to understand universe. So it is a story of life in context of origin of earth and universe. Considering the size of the universe, earth is just like a or dot. Universe formation, age 13, and it was explained by Big Bang theory. According to that, a single huge explosion caused expansion of matter, temperature came down, and yes, within the universe there are many galaxies. They are nothing but clouds of gas and dust which got condensed due to gravitational force. One of the galaxy, Milky Way galaxy, of which Earth is a part, and Earth was formed 4 at the time of Earth formation. There was no atmosphere. Okay, actually, molten mass, methane, ammonia, etc., inorganic substance were there. Hydrogen and helium were formed later. Ozone was also not there in the beginning. UV rays split the water molecule into hydrogen, and hydrogen being lighter escaped to the surface and formed atmosphere. That's why primitive atmosphere is called reducing atmosphere, because of abundance of hydrogen. There was no free oxygen. Oxygen reacted with methane and ammonia to form carbon dioxide, water and other compounds, but no free oxygen at that time. Next, life appeared 500 million years after the origin of Earth. Non-cellular forms. 3. First cellular forms. 2 billion. Okay. And actually, the origin of life explained by different theories. Theory of panspermia states that life came from outer space in the form of, it is still a favorite idea among astronomers. Then spontaneous generation was followed for a long time, which state that life originates spontaneously from decaying, rotten matter, non-living thing. It was disproved by Louis Pasteur. Louis Pasteur showed that pre-sterilized flask, life forms did not appear from the killed yeast. But in the flask which was open to air, life forms appeared, and concluded that life comes from pre-existing life and disproved spontaneous. But it does not explain how first life. Who proposed? Next one. Oparin and Haldane. Oparin and Haldane, they proposed chemical evolution followed by biological evolution. They believed that first inorganic molecules reacted to form organic molecules, and these organic molecules gave rise to first life forms. That's the central idea of that chemical evolution. It was experimentally verified by S.A. Miller. They created simulation. Yes, similar conditions of primitive earth in the lab, and under the simulated condition they verified it. What was the reaction mixture? Ammonia, methane, hydrogen, water vapor. Temperature was 800. Energy was provided in the form of electric discharge. Okay, and when the experiment was run for few days and they collected the products, analyzed the sample, they found that organic. They used inorganic, but analysis showed simple organic molecules, amino acids, first form, alanine, glycine, aspartic acid. Later, these experiments were conducted by others. They obtained pigments, nitrogen bases, sugars, and all. These experiments proved that life came from first organic, which in turn developed from inorganic. And another thing, analysis of contents of meteorite also showed same product, indicating that same type of changes are happening somewhere else on the other planets. Next one. Now, first form, non-cellular forms, they were nothing but aggregates of giant molecules, RNA, proteins, polysaccharides, etc. Remember that point? And they could reproduce, multiply and give rise to new molecules. And they are called protobionts before becoming the life forms. First formed cells were anaerobic heterotrophs. Gradually they evolved into chemoautotrophs. Chemoautotrophs gave rise to anoxygenic photoautotrophs, bacterial chlorophyll. Later, oxygenic photoautotrophs. With that, there was evolution of oxygen, reducing atmosphere gradually transformed into oxidizing atmosphere. Aerobic forms also evolved. First formed single cell, from there multicellular forms evolved. Then comes special creation theory. Special creation theory was a religious theory. It was accepted till the middle of 19th century. According to that, all life forms were created by God. And there are three connotations. All the forms that we see on the earth were created as such. Since the time of creation, they remain the same and will remain the same in the future also. Earth is only 4,000 years old, not millions as suggested. These are the connotations special. Later, Darwin. Darwin traveled HMS Beagle 5 years, visited various continents, collected data about plants and animals, was fascinated by finch birds on the Galapagos Island, and after observing all this, he stated that all living organisms share similarities to different extent, not only among themselves but also with the organisms that lived in the past, which indicates common ancestry or suggests the evolution, not creation. And not only that, some of those forms which lived at earlier may not be living now, they have become extinct. Some new forms appeared in the course of time. So, evolution idea was given by Darwin. And Darwin believed that there are built-in variations in every, and whenever there is a competition or any problem, variations which are useful to the organism to survive in that competition, it has better chance to survive, said to be having fitness. According to Darwin, fitness is reproductive fitness, because organism which is having fitness to survive, definitely it survives, reproduces and passes on the trait to leave more number of offspring. So, reproductive fitness. Okay. And yeah, that is Darwin's idea. Around the same time when Darwin proposed his natural selection theory, Alfred Russel Wallace also, working at Malaya, he also came to the same conclusion, and there is a concluding point. Age of the earth. Organisms lived at different time periods, and time periods were divided eras, periods, epochs, etc. Geological history correlates with biological history. Next one. Evidences. First one, paleontological. Evidences are available in support of evolution from almost all fields of biology. Paleontological evidences, fossils. Fossils are the remnants of the organisms that lived in the past. Most common site, sedimentary rocks. That means when animal dies and around the same time if sedimentation occurs, layer formation occurs, automatically it gets trapped, its body gets trapped in that layer and may remain undisturbed and available as fossils sometime later. So thus fossils are found, and if you arrange all these fossils in different strata, you can understand the sequence of evolution also. Fossils in the top layers indicate recent fossils, bottom layers, and you can see a gradual change in the organisms also in. And how to find out age of the rocks and fossils? Carbon dating technique. Half-life period is used to measure. Next, what is the example here? Archaeopteryx fossil with both reptile character and bird character clearly suggests evolution of birds from reptiles. It's a fossil, it's no longer living, Archaeopteryx. Then next one, embryology. Von Baer, he observed that all vertebrates develop paired gill slits behind the head, but gill slits are functional only in adult fishes. And he stated that all these vertebrates are repeating the characters of adult fishes. What is the theory called? Recapitulation, biogenetic law, ontogeny repeats phylogeny. But this was disapproved by Van Baer. He stated that embryos never pass through adult stages of the ancestors or other organisms, and he developed the principles of embryology also. Next comes morphology and morphological anatomical evidences. Morphology and anatomy. First example, cheetah, whale, bat, human, four limbs. They are performing different functions, flying, swimming, running and all that. But basic structure is humerus, radius ulna, carpus, metacarpus, phalanx. Why different organs having same internal structure, same anatomy? Because all these evolved from, because all these are mammals evolved from common ancestor. That's why they are having same internal structure even though they are adapted for different functions. Homology indicates common ancestry, and why they are adapted to different functions? It indicates divergent. So homologous organs are a result of homology indicates. Other examples, mouth parts of insects, brains of vertebrates, hearts of vertebrates, bougainvillea and cucurbita, thorns and tendrils. Different unrelated organisms developing similar characters to live in the same habitat subjected to same selection pressure is called analogy. Such organs are analogous and they indicate convergent evolution. Wing of insect, wing of bat, unrelated, but both develop wings to fly in the air to live in the same habitat. Other examples, flippers of penguin, flippers of dolphin, eye of octopus, eye of mammal, sweet potato, root and stem modification. Function is same, storage, but different structures. Analogy. Morphology, anatomical evidences. Then biochemical evidences. Different genes and proteins of different animals performing same. Genes and proteins in different organisms performing same function. Why? In different organisms, why should they function same? Because all evolved from common. That means it also indicates common ancestry. Another evidence. Man, by careful selective artificial breeding, developed many breeds of dogs, animals and plants for horticulture, agriculture, sports and security, etc., dogs, etc. If man could create so many new breeds in a span of hundreds of years, couldn't nature have created that over millions of years? So that is other supporting evidence. Then comes anthropogenic evolution, no? Industrial, okay. Industrial mechanism example for Natural selection. Biston Betularia moth. In year 18, survey was conducted at a place in England, and that time white moths were more because that time lichens were abundant on the tree trunks, no pollution, and white color provided better camouflage. They survived more in number. After industrial revolution, lichens cannot grow in polluted areas. So, plus soot and smoke released from factories and industries coated the surroundings. Surroundings turned dark. Under those conditions, black color melanic formed better chance of survival. So their number increased. So melanic forms are favored after industrialization. That's why it is called industrial melanism. Nature selected the ones with better camouflage. No variant was wiped out. Both are there. The proportions changed in that. Next one. Anthropogenic, excessive usage of herbicides, pesticides caused resistant varieties. Not only that, excessive usage of antibiotics, antibiotic resistant bacteria, excessive usage of drugs, drug resistant eukaryotic cells and organisms. All this is happening within short or lesser time scale because of excessive usage. Without excessive usage also they may develop. But for that to occur, millions of years time is required, but we are speeding up this process by using them excessively. Conclusion. Evolution is not a directed, it is stochastic because it occurs by chance. Next one. Adaptive radiation. Appearance of different species, many species within a geographical region starting from a point radiating in different habitats or regions of the geographical area is called as adaptive radiation. First example, Darwin's features. Original one, seed eating. From that, different beak varieties evolved on the Galapagos. Same geographical area, Galapagos Islands, they evolved in different types. So, best example for adaptive radiation. Marsupial evolution in Australia, different types, adaptive radiation. Placental mammals evolution in Australia, adaptive radiation. More than one adaptive radiation occurring in the same geographical area results in convergence, and you know that only those members in those pairs are to be considered for convergence. Flying phalanger, flying squirrel. Spotted cuscus, lemur. Only these pairs exhibit convergence. These exhibit adaptive or divergence. These exhibit adaptive radiation or divergence. These pairs only exhibit. Okay, next one. Biological evolution. Rate of evolution can be linked to lifespan of the organisms, and it is believed that natural selection process must have started in the beginning stages when first time cells with variation in metabolic activities appear. Whichever metabolic process more efficient, that must have been selected by nature. So process must have started that time only. And regarding this time span, bacteria, they multiply very fast. Number reaches millions within short time. So remember that example. In a bacterial colony, sustaining on a particular feed, if feed is changed, only few members which can utilize a new feed, they survive, reproduce, pass on the ability to next generation. So they number gradually. Majority which are not able to utilize a new feed, they die. So gradually after some generations, A decreases, original variety. B forms increase, and we say simply B evolved from A. And this evolution of B variety from A can occur within days because they multiply very fast. For the same type of evolution to occur in higher animals, it may take millions of years time. So it is linked to the lifespan. More the lifespan, rate of evolution is. What are the key here there is important sense usually statements. Adaptability is, it has a genetic basis, because see in this example few members, B members, They could utilize new feed. That's why they survived and passed on it next generation. That means it is inherited. That ability is inherited, genetic basis. And they concluded that fitness is the end result of ability to and get selected by nature. Two key concepts of Darwin's theory. Before Darwin, who explained evolution? Lama. But he believed that it was due to use and disuse, and he gave example giraffe, using the neck and forelimbs, causing long neck giraffes and all that. Remember those examples. Uh, who disproved Lamarck's inheritance of acquired characters? August Weismann. Decaudalization experiment. Cutting the tail. But in 21 generations, single rat would not appear without tail. And he provided explanation also. We talked about ear boring, nose boring, Chinese women feet, so many examples. In all the cases, somatoplasm, somatic cells are involved, which do not take part in reproduction. Only changes which occur in germ plasm alone will be passed on to next generation. That is called theory of germ plasm, proposed by August. Who proposed Pageant's theory? That was also disproved by the same experiment. And then Darwin's, again, there is no conclusion about this. Evolution, natural selection, process, or end result. When you take the story of the world, evolution is the process. The world we see with the animate, inanimate evolved. So evolution is the process. When you talk about story of life forms on earth, they evolved through natural selection. That means natural selection is process. Evolution is end result. But still they do not conclude there. Still we do not know whether they are process or some other process which are not known to us caused this. Okay, next one. Darwin's theory. Darwin was influenced by Thomas' essay on population, and his theory is based on some observations which are facts also. Organisms always trying to reproduce maximally, but resources are limited, and every population shows variation. Superficially they may look similar, but there are variations which play important role in the struggle for existence. And he talked about struggle for existence and survival of the fittest. And when organisms are selected, or those which are having all these points. For example, natural resources are limited, populations are stable in size except for seasonal fluctuation, members show variations in characters, though they look same superficial, they have variations, and variations are inherited. So theoretically every population can grow in size, but practically the problem is resources are limited. So at one or the other point, competition for resources increases. In that competition, organisms with variations, better variations only survive. They have better chance to survive, reproduce, leave more number of progeny, pass it on to next generation. And that is said to be selected by nature. And when this happens over generations, gradually a different looking population appears, and we say that so and so population evolved from the pre-existing one. That is a central idea of this Darwin's theory. But there are some drawbacks. After Darwin, Hugo Debris proposed mutation theory to explain evolution. At the time of Darwin, Mendel proposed his principles but kept silent because he was not able to understand all that statistical data. He did not explain the origin of variations, and he had no idea about inheritance of the variations. Okay, Hugo Debris worked on Evening Primrose, Enothera lamarckiana, and he believed that evolution occurs only by large scale variations like mutations, but not small variations talked by Darwin. According to De Vries, mutations are sudden, random, directionless. Darwin's variations are small and directional. Plus, according to Darwin, evolution is a gradual process, slow process, because it occurs by minor variations. According to De Vries, sudden in a single step or saltation. Suddenly, new species appears according to that. Later, this genetic studies, population genetics provided us more information, that is hardy. Sum total of genes in the population called gene pool. According to Hardy and Weinberg, gene pool remains constant or gene frequency remains constant generation after generation. Such a population is said to be in a state of equilibrium. What is the formula for frequency? Number by total. You can calculate allelic frequency or genotype frequency. Remember the calculations. Hardy-Weinberg equilibrium is maintained only in large population undergoing random mating and absence of all those factors. Natural selection, mutation, all these factors should not occur because they disturb the equilibrium. And any deviation from Hardy-Weinberg expected values indicate that population is evolving. More the deviation, more is the evolution. No deviation, no. Or less deviation, less. So this mathematical expression helps us to understand the extent of evolution. Hardy-Weinberg equilibrium is disturbed by 5 factors. What is first one? That is when organisms migrate from one population to other. Along with them, some genes are also lost. And if it occurs multiple times, it is called gene flow. Disturbances occur in both the population. Changes occurring just by chance. Genetic drift. Genetic drift effect is high in small, isolated populations. And the 2 forms of genetic drift are bottleneck and when some members get drifted away from the original population, settle down somewhere else, they become founders for the new population and gene frequencies will be decided by founders. So it is called founders effect. And bottlenecks are natural calamities. Whenever natural calamity occurs, all organisms die. By chance, few may survive and the frequencies of next population or subsequent decided by survivors of bottleneck. Who survived with that bottleneck. That's why it is called as bottleneck. And then mutations. What is the statement? Pre-existing. Microbial experiment showed pre-existing advantageous when get selected result in the appearance of new people. So whenever mutations occur and the mutation is useful, it will be selected and new phenotype appears in the population, which can cause change in the Hardy-Weinberg equilibrium. Plus during gametogenesis, meiosis crossing over occurs, resulting in genetic recombination. That also can cause disturbance in the equilibrium. Last one, natural selection. Actually, in the population, changes occur due to migration, genetic drift, mutations, recombinations, and once variations occur, the one which is having more fitness, it would be having more reproductive success and that gets selected. So natural selection makes this population look different from original population and we say new species is evolved. Type of natural selection. Remember, recently that question, different examples are given. Always remember, whatever is the average phenotype, if the same thing is maintained afterwards. So, majority acquired the average phenotype. Second one, if a new phenotype, different phenotype appears, that means not the original one, another character is selected. Then it is directional. From less milk yielding variety, high milk yielding variety. From original white moth wing population, white moths, black moth population number increasing. So one to another character, that is directional. At a time, two or more getting selected, then it is disruptive. Majority acquire peripheral character value other than the mean value, that is disruptive selection. What is the next one? Animal, more important. First, early reptiles, synapsids, pelycosaurs, therapsids, mammals, and then sauropsids. Some sauropsids gave rise to thecodons. Thecodons gave rise to birds, crocodiles, dinosaurs. Dinosaurs became extinct. And all other sauropsids gave rise directly to chelonians, lizards, snakes, and all that. First unicellular forms or first cellular forms appeared. Two, how the non-cellular aggregations without cell membrane transformed into first cell membrane, exact mechanism is not known. Unicellular members gave rise to multicellular, and first formed unicellular members, they had the ability to split water, release oxygen, same as splitting of water molecule by photosynthetic pigment. Same mechanism must have operated, and they evolved into those forms. Then numerical data. 500 million years, invertebrates were formed and active. 350, jawless. 320, seaweeds and aquatic plants. Low-pinned fishes, around 350 million years ago, they started moving between water and land, and they gave rise to the first amphibians. First formed amphibians became the present amphibians that we see as descendants of those. Then amphibians gave rise to reptiles. Reptiles are more successful. They lay thick-shelled eggs which do not dry up, unlike amphibians. And they are also first formed reptiles no longer living. We see their descendants only. Reptiles dominated the earth for about 200 million years. And around that time, gymnosperms, no, pteridophytes, they were there, they fell down and formed cold deposits. Some of the reptiles went back to water to form fish-like reptiles, ichthyosaurs. Among the terrestrial members, largest is Tyrannosaurus rex, 20 feet height, fearsome, huge dagger-like teeth, and 65 million years ago, suddenly they became extinct due to climatic changes, or some believe that they evolved into small sized birds. Truth may lie between these two. And once reptiles came down, the world was taken over by mammals. First formed mammals were shrew-like animals, and viviparous, protect their young one, more intelligent. And there again we have this continental drift examples. So, one is South America earlier had animals like rabbits, bears, hippopotamus, horse, etc. But due to continental drift, once South America joined North American fauna. Second example, Australia is a land of marsupial. They survived in Australia because of less competition with other mammals, because it got isolated due to continental drift. Then humans more intelligent, self-consciousness, language skills, we talk about all that. Last one, human evolution. Remember? Dryopithecus and Ramapithecus, 15 million years ago, more man-like Ramapithecus, more ape-like Dryopithecus, and after that 3 to 4 million, no name is there, human-like primates walked in grasslands. They walked and upright posture. Then 2 million years ago, Australopithecus, and among where? 3, East Africa. 3 to 4 million years ago, primates, Ethiopia and Tanzania. And among the bones of Australopithecus, some bones looked different. Homo habilis. First human life 650 to 800. Cranial capacity hunted with Australopithecus hunted with. And both Australopithecus habilis did not eat. Next, 1891 Java or 1.5 million years ago, Homo erectus, completely erect body, 900 cc, ate meat and also made fire first time. Then comes Neanderthal man. Neanderthal man, 1 lakh to 40,000. Central and East Asia and practiced 1400 cc plus covered the body with the hides and practiced some rituals, burying the dead, etc. Then comes Cro-Magnon man, 1650, lived in caves and started cave painting, used to make many ornaments and all that. Last, and one more thing. First Homo sapiens arose in Africa, moved to different continents, evolved in different races. Modern man evolved during Ice Age, 75,000 to and 18,000 years. Beambetka. Agriculture started 10,000. Okay. Zoology preparation day, try to complete all the syllabus one go. Okay. Afterwards, you won't be having sufficient time to revise. You will be having almost every day grand test. And during these tests, what you can do is, suppose after writing the exam, okay, suppose you made 3-4 mistakes. Immediately check those subtopics where you made the mistake. That means every day after you make mistakes, you will be revising few subtopics. Is that clear? And it is not only about zoology, you have to do it for all the subjects. Definitely, it takes time. So try to complete few topics every day, especially those topics where you make mistakes. By that way, some topics will be revised in course of time. Okay, all the best.